for joining us today, and also thank you to thank you for to the people that is watching us on the YouTube channel of Leitat that are watching the stream. Sometimes it doesn't work. Yeah, the other one. Let's say, yeah. Okay, so there are around 7,000 rare diseases identified today, and only 5% of them have treatment. So that gives us an idea of how important it is that researchers all over the world focus their research on a better understanding of those diseases and finding, helping us to find new treatments for help those patients that sometimes has a very little life expectancy. Leitat is part of the European project Bad Cure that is focused on the Batten disease that the Dr. Sarah Moll will explain you later. And we wanted to take advantage of the opportunity of having here all the members of the consortium to share with the scientific community all the results that we have obtained in this project over the past 3 years. And also we wanted to invite other researchers and clinicians, experts on rare diseases, so that they can explain also what have been doing in their current research. So we will have 6 speeches today and a coffee break in the middle. And that's it. Thank you, and I hope you enjoy the afternoon. Our first speaker is actually Dr. Sarah Moll, that is the group leader of the Laboratory for Molecular Cell Biology in the UCL, the University College of London. Thank you, Sarah.
So I use this, okay. I'm just debating whether to stand here. Camera, am I better standing here or up there? Here. Okay. I won't know what slide's coming next, but that's okay. So then I just need this. Okay. Okay. Right. So what I thought I would do is talk for about 20 minutes or so on telling you about Batten disease, telling you about the work that we've been doing as a consortium, sort of working together to try and understand and develop therapies, new therapies for Batten disease. So just some pictures to introduce you to the consortium. So first of all, we're an Horizon 2020 funded consortium. We consist of 14 partners from 7 countries, and you can see those coloured there in Europe. And that's it. So this is us at the beginning when some of us didn't know each other very well at all. A picture of us mid-meeting looking much happier and more relaxed, and then that was last September at the end of an international meeting, and we have our final meeting here in Barcelona. So as you can see, quite a number of us. So the whole concept of the project that we've called BATCure is to work along the pipeline of developing that's required to develop therapies. Now, we'll tell you a little bit more about Batten disease in a moment, but there are a number of different types, and we chose to focus on 3 types in particular that are caused by mutations, defects in membrane proteins, intracellular membrane proteins. So these are the most challenging types to develop therapy for within the BATCure field. So so the concept, as it were, was to spend quite a lot of our effort in terms of sort of the pre-discovery work that's required. So trying to understand more about the disease in animal models, in cells from patients, and also in very simple models that we developed. And also in patients themselves. And then our focus was to, we decided to focus on developing drug and gene therapies. So so we already had some leads, etc. We needed to optimise and just further that whole process. So there's been a whole package of work around that. And then we also wanted to be aware of anything we could do in advance to prepare ourselves so that when we reach the stage of clinical trials, we could get to those as soon as possible. So that's what the last section was for. So, as I say, we've sort of the fairly standard pipeline in terms of translational research is listed here, some from our disease models through to identification of good markers to follow the disease and therefore to assess the efficacy of therapy, and then our lead development, optimization of current leads and drug and gene therapeutic strategies. So the different groups within the consortium already had some leads, so some small molecule leads and some background work on gene therapy. So whilst some of the aspect, we were starting here, other aspects of the biology sort of fed in partway through, and the thinking behind that was that we would make better progress building on things that we'd already gained quite a lot of knowledge about. Right, I'm not expecting you to be able to read this, but just simply to say that with a large group of people and having some quite complex deliverables and milestones required for the EU project, that we had to be fairly ordered, reasonably ordered about it, very ordered about it, and mostly we've kept to the time. There have been some deliverables that have been delayed for technical reasons, etc. But mostly this was a real help to sort of keep the project moving forward. And of course we have an organisational structure to try and work out exactly where all, so that we can work along all the way all things fit in. So anyway, I shall move more onto the science now. So this particular consortium is actually one of a number that have occurred over the last 25 years across Europe. So there's a strong history of collaborating across Europe for Batten disease, and whatever happens with the UK and Brexit, hopefully that will continue. So this is quite an early one. So that's me at the beginning, looking much younger than I am now. That was NCL models, where we were developing some of the very first models. And then there was a DEMChild led by Angela Schultz here, which continued that work and had a real focus on setting up a disease registry for Batten disease. And then there's Here's Backcure with a translational focus. And along the way, we've produced a number of books as well, which we class as reference books. So we've worked very hard to develop a good resource for anyone who comes into contact with Batten disease that needs something to dip in and out of. Such a rare disease. There's a lot of knowledge out there, but it's spread. And of course, many people don't know too much about it. And by the end, or beyond the end of Backcure, we'll hopefully be producing the 3rd edition of this book as well. Okay, so back to the disease now. So Batten disease, its long name is the neuronal ceroid lipofuscinoses. It's a family of diseases, usually childhood onset. And the Batten comes from this gentleman here, Frederick Batten, who was a physician in London around the turn of the last century. So it's one of the most frequent neurodegenerative disorders of children, and in the UK, around 10 new cases a year. And it's difficult to know across Europe, but I've calculated that would make about 70 new cases across Europe each year. So these diseases have a characteristic ultrastructure of the storage material that builds up in the lysosome, which is also autofluorescent. And there's just examples of one type here, which has a fingerprint profile under EM. Each Batten disease is clearly inherited disease, clear monogenic inheritance, and all except one are autosomal recessive. And at the moment, these diseases are incurable. So, what I'm going to do now, so the most common type of Batten disease is one called CLN3 disease, juvenile CLN3 disease, which begins with quite a rapid visual loss around the ages of 6 to 10. And then sort of gradually disturbance in speech, onset of seizures, and then a progressive decline of cognitive and motor functions, and also accompanied by psychiatric signs, sleep disturbances, extrapyramidal signs, and eventually leading to death. and this could be in the late 20s. So here are two patients with juvenile form up here. So Laura, who's at the very early stage, and then this gentleman who is more advanced. One of the more rapid types is CLN1 disease. So here's a girl who is at 1 year, looks perfectly healthy, but 6 months later she can't even sit up without being helped, and a few years later is very severely affected. This is a group of families with their children, and most of these children will have CLN2 disease, which has an onset between 2 and 4 years. So over the years, and this was my background, we have identified quite a number of genes now that cause Batten disease. So in the beginning, of course, these were the most common types, and then more recently these have been genes identified in single families. And along the way, I set up a mutation database, which if any of you do come across Batten disease patients, you might find helpful. So this is hosted by UCL. It's part of a bigger resource website, but in terms of the databases, these are tables put together for each gene, summarising patients or the mutations separately, and they're trawled from the published literature or information supplied by diagnostic labs, or even sometimes families themselves. And we try and present the data in a they're just Excel tables, so you can download them and manipulate them how you choose, and it can be helpful, for example, looking to see what mutations are particularly common in a country that you might have to consider in terms of diagnosis. However, what has been extremely useful from this is that when we look at all the mutations and the phenotype associated with those, that we see really wide disease phenotype variation, as of course is increasingly recognised with many diseases now. So just for example, looking at the CLN10 gene, which is the extremely early onset, very rare, so complete loss of function of this gene, which encodes cathepsin D enzyme, has a congenital onset, but there are also cases with mild mutations where onset may be delayed by a few years, or even up into sort of late teenage, early adulthood, and that applies for most of these genes. We also have a few mutation-specific phenotypes, so particular mutations cause a disease that wasn't initially recognised as an NCL. So, for example, CLN8 gene, the first mutation, or rather the gene was first identified in a group of patients with a disease called progressive epilepsy with mental retardation, or northern epilepsy, which wasn't considered to be an NCL at that time. And then very recently, we're now seeing in quite a lot of the genes some very late onset disease. So CLN3, which I'll refer to later, we're now seeing adult patients who are only blind, and their mutation is found in CLN3. So this phenotype, genotype-phenotype is really broadening. Right, so number of genes. Is there a common theme? So this is just a schematic to show you where the location of the proteins encoded by these genes are, and you can see that there's quite a few genes that encode enzymes within the lysosome, so or even transmembrane membrane proteins of the lysosome. So that fits with the storage material that accumulates in the lysosome. And then, but we also have a few genes that are encoding ER membrane proteins. So somehow there's a connection in this disease between what's happening in the ER and the lysosome. Now, whether that's via a trafficking route or whether it's via direct organelle contact, we're not too sure yet. Okay, so back to BATCure, and what I thought I'd do now is give you some examples of some of the work that we've produced through this project. So we had one work package that was providing new models and tools, and then a lot of interconnected work packages really focusing on, as I say, the biology and the basic background to the therapeutic development. So just to dip into some of these work packages. So work package 1, which was called New Models. There are quite a lot of model organisms available for Batten disease. Some of them are engineered, some of them are naturally occurring. So for example, there are naturally occurring primates and sheep and dogs that have mutations in genes that cause Batten disease in humans. But there are also some lab models as well. And within BATCure, we chose to focus on yeast and zebrafish and mice. So yeast as the simplest organism possible to study the genes that are conserved down to yeast. A few of them are for Batten disease. Zebrafish as a simple vertebrate that allows for some very fast screening or testing of drugs and also genetic manipulation. And then the mice for developing the gene therapy and testing the effects and also understanding more about the disease. In terms of developing new tools, LATAT here have been working on antibodies. It's been quite a challenge, but we have some. And we also have a lab that was developing iPS cells as well. So that sort of was the basis of the model work package. Now, the second work package, Pathway Lead, was an enormous work package coordinated by Juan, who's sitting right at the back there. So we always had to give a lot longer for any presentations within this work package. So I've tried to summarise it very briefly here. So what we were trying to do within this work package was understand more about the basic biology and gene function to provide in lots and lots of different ways. So genetics, cell biology, metabolic approaches, proteomic and bioinformatic. Then from that work to identify specific targets that we thought might be responsible for some of the defects that we were seeing within neurons in the disease, and then also to use that information to look at the effect of new compounds or test compounds that we might be studying during the process of the project. And obviously this work package was receiving information from some of the others, so of course from the new models developed, but also generating information that went out into other work packages. Excuse me. So just to highlight some of the work we've done using yeast as a model organism, because this is quite unusual if you're looking at a brain disease to think about using yeast for this. And what we did here was to try and do a very sort of take a systematic approach and look and identify genetic interactors with the conserved gene in yeast. So the CLN3 gene is conserved down to yeast, highly conserved, and we know that yeast lacking the equivalent gene, which is called BTM1, they have a plethora of phenotypes and they can be rescued by expression of the human gene. So what we did here was do a synthetic genetic array approach where we deleted all the non-essential genes in yeast in the background of a yeast also lacking the equivalent CLN3 gene. And then there are a number of approaches we can take with the hits that we find. So we can look for differences between the different strains that we used. We also compared different disease-causing mutations as well. So we had quite a lot of information to deal with. We can look at things that overlap between them and significance, strongest interactors, and things that are common between them. So of course this is a lot of work and will continue beyond the end of the project. Now, in terms of work package 2, and trying to understand about the metabolic reprogramming that became apparent in the CLN7 form of Batten disease, where there was clearly some damaged mitochondria and some metabolic rewiring that was going on. So this is work that Juan is unpacking and is very interesting, and of course may help us in terms of understanding which drugs are going to help the CLN7 type of disease. Another lab from Germany, led by Stefan Stork, we're looking at the disturbance of lipidomics and proteomics, and here just showing the results they've generated for the CLN7 type. So looking at sort of depletion of quite a number of the soluble proteins within the CLN7 lysosome. They've also found impaired mTOR reactivation in fibroblasts from the mouse model compared to the wild type, and impaired tubulation from the autolysosomes in the CLN7. The CLN7 gene, if you remember, is one of the lysosome membrane proteins. And when they look in the lysosome, either you're looking at the brain or, again, using the mouse fibroblasts, they can see an increase in certain lipids, and they can see, again, the decrease in the enzymes within the lysosome. So starting to understand changes, and some of these changes, of course, are going to provide useful biomarkers. When we have treatments, we'll be able to see whether the treatments restore the level back to what we expect for wild type, or whether actually it may indeed rescue, but it may produce a new normal. Okay, so moving on to sort of thinking about the drugs and development of drugs. We took two approaches for screening for compounds. One was screening, this is a lab in Italy, led by Diego Medina, used human cells and did high throughput screening on human cells. And, we also, again, made use of the yeast and did high throughput screening to identify compounds that rescued yeast, and again, we can of course test yeast that model different mutations in the disease gene, and then also test the compounds on yeast that model other diseases, other lysosomal diseases beyond Batten disease, and then of course take those compounds into patient fibroblasts and eventually into testing in vertebrate animals. So in terms of therapy, as I say, we decided to focus on gene therapy and drug approaches, and we're aware that we have to target the brain, therapy to the brain, to the eyes, and then we also have to think about the rest of the body, because the storage material occurs throughout the body, although the disease that we recognise is really a brain disease. But of course, if we're able to improve what's happening in the brain, it may be that other problems arise during the rest of the body, in the rest of the body. So to show you some of the gene therapy work, we've first of all been developing eye gene therapy, and we began with CLN6, and just very briefly, in a mouse model that's untreated, at 9 months, the photoreceptor layer, which is here, is severely reduced. So from there, the number of photoreceptor rows is very severely reduced, but with a gene therapy, we can restore the rows of photoreceptors here, which you can see visually here. But what was really interesting about this was in all other eye gene diseases that have been targeted by gene therapy, if you put the gene into photoreceptor cells, that's what worked. You stop the photoreceptor cells being lost. But for this one, if we did that, the photoreceptor cells still died. We actually had to target the gene therapy into the middle layer of the eye in order to prevent the loss of the photoreceptor. So this was a novel finding completely, and obviously we've now been aware of that as we've gone forward with some of the other gene therapies for the eye, for the NCLs. And then here, Ahad Rahim has been leading on gene therapy for the brain, and again, just to show you one result of the CLN6 mice that would normally die by a year, but with the gene therapy into the brain, we're extending the treatment to 2 years, which is about the length of the same as wild type mice. So this is working very well as well. Okay. Now, if we're going to move forward with therapy, you do and clinical trials on rare diseases the approach that's been taken is to compare the effect of any therapy against the known natural history of the disease, rather than having a control group that isn't treated at all. And the reason we can do this for Batten disease is because of the Child Disease Registry, which I mentioned earlier, which has now become an international disease registry where a lot of information is now collected for all the different types of Batten disease. There have been a number of clinical trials over the years for the NCLs, most of which have had little benefit at all. There's just one that's been successful. And in terms of the three that we're working on, the CLN 3, 6 and 7, there have been some very recent trials started in the USA with gene therapy where they're really just trying it out. So it will be years before we know the result of this, but what we feel we're doing within our mouse gene therapy work is really providing some of the science behind gene therapy to really understand what's going on and what the key things are that we need to be aware of. Okay, just one of the highlights from the consortium was we ran an international meeting last year in London, and so there all the people that sort of gathered from around the world working on Batten disease, and yeah, that was a good outcome for us. I've already mentioned the fact that we want to produce the 3rd edition of the textbook. And I'll close by just showing you all the different logos of the labs that have been involved in this disease, in this project. And I'd be very happy to take some questions. Okay.
Thank you very much, Sala.
Yeah.
If anyone wants to raise any question regarding this project or something like that. No? I can.
Continue in Spanish. I don't speak
I can translate.
Yeah. Yes, okay,
Yes,
thank you.
No problem.
No, yo soy Gustavo, soy el papá de Marta y María, que tienen una enfermedad, bueno, tienen Batten CLN6. Sí, tienen Batten CLN6 y están participando en el ensayo clínico que hay en Estados Unidos actualmente de terapia génica. Con la doctora Emily de los Reyes muy bien. Entonces, pues bueno, la mayor lleva 1 año en el ensayo clínico y la pequeña 6 meses. Quería preguntar pues, 2 cosas. La primera es si todas las vías de investigación que se están llevando aquí a nivel europeo pueden ser complementarias con esa terapia génica que están pues eso, tratando a mis hijas allí en Estados Unidos. Y la segunda pregunta es, no sé, es muy complicado encontrar a familias que están en nuestra misma situación, sobre todo pues con la enfermedad del tipo 6 que tienen nuestras hijas. En España no hemos encontrado, hay unos gemelos que la tienen, pero no hemos podido contactar con ellos y no conocemos de nadie más. Thank you so much.
Yes.
I can help you with the second question about the
Yeah.
Okay, so first of all, to answer your question about the therapy. So the principle of the gene therapy in the States and what we've been working on is very similar. And we've been trying to, we're trying to think about how early you need to start this therapy, thinking about dose, thinking about sort of the details of it. So going beyond what was known before the trial started. And so I guess all we can say is certainly for in mice, which he will know about and which was done in the States too, that in mice the gene therapy seems to work well. But of course we don't know what it's like in a child, a larger animal, you know, or a child. Okay. Okay. So, yeah. So in terms of getting in touch with families with the same mutation, a lot of that seems to be done through social media now. But of course, we will know from the mutation, we can look in the database to see approximately how many families we're aware of and where they're from. So that could help target families perhaps, i.e. is it a Spanish mutation or is it throughout the world?
Solo decirte que en España tenemos dos grupos de neuropediatras que estamos trabajando con enfermedades lisosomales y estamos haciendo una base de datos y estamos recogiendo todos los pacientes con los diferentes grupos, entre ellos las lipofuscinosis, pero que no siempre todas las familias quieren contactar con otras familias y eso es algo que nosotros también tenemos que respetar. Nosotros siempre les decimos que hay posibilidad de contactar con otras familias, la mayor parte de ellas están de acuerdo. Bueno, algunos no lo están tanto, ¿no? Y sobre todo los que viven en sitios más periféricos o que no van a centros de referencia todavía, esos son más difícil de contactar con ellos. Por eso estamos haciendo ahora un trabajo de búsqueda con todos los médicos neuropediatras españoles. Sí, luego.
Nosotros estamos haciendo distintos eventos para recaudar dinero, pues para ayudar en la investigación. No sé si sería posible el poder también pues contactar con ellos para pues bueno, si lo necesitan, si es viable. Ajá.
Sí, podemos, en ese caso, en la pausa, 4 menos cuarto, podemos hablar y coordinarlo todo, porque de hecho el foro es el adecuado. O sea, aquí hay varios investigadores de varios centros, de varios hospitales. Creo que es el foro adecuado y conseguiremos una forma a ti. Okay, any more questions? I think we have some for 1 or 2 more. If not, I think we are in an excellent time, so we can move on. So I'm so happy to introduce Dr. Martinet from Minorix Therapeutics. He's the CEO of this of Minorix. This is a company developing therapy for I can remember the names of
ALD. I will talk about it, don't worry. Okay, so thank you. It's a pleasure to be here. I founded this company a few years ago with the goal to develop therapies for orphan CNS conditions with high unmet medical need. Today, we are leading the space with MIN102 in a particular disease called adrenoleukodystrophy, or ALD. We are also moving into other indications with the same molecule, such as Friedreich's ataxia, and I will talk about these two diseases. And we're also preparing to move into additional conditions with the same molecule, because of based on the mechanism of action, it has a potential to go beyond these two indications. We are a very small company located in Mataró, north from here, founded back in 2011, and we have most of the team here in Mataró, but also have some people in the south of Brussels, in Charleroi. This is just a snapshot of the clinical trials that we have, or the development that we have in development, mostly ALD and Friedreich's ataxia. And as I said, we are preparing for moving into other spaces. So to start, let me talk a little bit about the molecule and why we think that this is a good candidate or good molecule for this kind of disease. Min-102 or leriglitazone is a PPAR gamma agonist. This is a small molecule. There have been several PPAR gamma agonists around, and some of them have been in the market already for diabetes. But the issue with these PPAR gamma agonists is that the lack of sufficient brain penetration to have sufficient levels in the CNS for efficacy. There was a pioneering work on the space of ALD and PPAR gamma agonists led by Aurora Pujol in IDIBEL here in Barcelona. They demonstrated that this mechanism of action could be used for ALD, but the issue of that particular molecule was to achieve those sufficient levels. And that's precisely with what we can achieve with Min-102. This is a molecule that reaches the brain at sufficient level and modulates the target at the required level for efficacy, at least based on what we know from preclinical studies. It's orally bioavailable, so it's easy for patients from that perspective. It's once a day after breakfast. And we use the molecule as an oral suspension instead of the classical tablet. And this is because we want to be sure that all patients are receiving the right level of compound. So we control or we monitor plasma levels for all patients, and we adjust, if necessary, the dose. The advantage of having a suspension is that we can also develop the same drug product, the same formulation for children. And that's also development that we are doing. In Friedreich's ataxia, we are enrolling children, and in ALD, there's also pediatric population, so that was the second key reason for developing an oral suspension. We have orphan drug designation in Europe and in the US for the treatment of XALD. We have IP, granted IP, and also the CMC is ready for multi-kilo scale-up. Because based on the stage where we are, pretty close to the market, all the CMC is another part that is less fancy than the biology, but it also needs to be pretty well controlled. The molecule does many things, and that's the reason why it works in such a complicated diseases where there are many things happening at the same time. It has an antioxidant and neuroprotective effect, not just in ALD or Friedreich's ataxia. This is in multiple conditions. It has a very potent anti-inflammatory activity, not only in the CNS, but in the CNS as well, if the compound is there. Again, this under different inflammatory stimulus, then the molecule is anti-inflammatory. It improves mitochondrial function. It has some of the pathways are very much linked with mitochondria. I will talk a little bit about pathways later on. It also ameliorates the lipid metabolism. That was one of the original reasons for the molecule. But in vivo, it improves the white matter structure in different models, promotes remyelination, and this is because of different reasons, because it improves oligodendrocyte differentiation, but it also protects oligodendrocytes themselves, and also improves myelin debris clearance, among other things. And finally, it delays disease progression in animal models, at least. Why ALD? ALD is our primary indication. There are some of historical reasons for that, but also we think that is a very good case to validate this biology in the CNS for this molecule. ALD is an orphan neurodegenerative condition. It's a pretty well-organized community, not as well-organized as some other rare diseases, but improving quite a lot. And since we started in this field a few years ago, this situation changed quite a lot. And there are some organizations at the international level, such as ALD Connect, which are quite powerful today. It has different phenotypes. To start, most of the patients have adrenal dysfunction. But if we focus on the cerebral form or the CNS-related, the most aggressive form of the disease is cerebral ALD. This can affect children or adults, but is typically seen in children, and death comes after 2-4 years from onset. So it's very aggressive and rapid disease, except if patients go through bone marrow transplant. In that case, this therapy can really stop the disease, or ex vivo gene therapy is in late development for this form of the disease as well, showing similar results as the transplant, but both are pretty aggressive for the patients in any case. Then there's the most common form of the disease, because most of the patients that are alive today have this form of the disease. Essentially, all patients reaching adulthood develop this form of the disease, starting at the 20s. is characterized by spinal cord related, is axonal degeneration in the spinal cord leading to motor dysfunction mostly, spastic paraparesis, so on and so forth, urinary incontinence, so there's several different things. But essentially this is affecting adults, men, but today we also know that women is also affected. This is an X-linked disorder. A few years ago it was considered to affect only male. Even in some of our slides at the beginning we were saying affects males. No, AMN is also affecting most of the women that are carriers. The difference is the age of onset, which in case of women comes much later, and the disease severity. Women do not develop cerebral form of the disease in most of the cases. So, and for AMN there's nothing at all. No treatment at all, and here is where we are the most advanced in the space. The disease is monogenic, so it's one single gene being affected. Is a gene related with the transport of very long chain fatty acids into the peroxisome for their degradation. When this is not functioning normally, there's accumulation. It's unclear if the accumulation is the trigger or is a biomarker or a consequence, but is there. And there are multiple pathways being affected, from the mitochondrial function, oxidative stress, inflammation, those same pathways that are modulated by PPAR gamma agonism. So that's essentially our target. These downstream pathways is what we are trying to correct in this disease. In ALD, we did multiple studies. Just to summarize briefly, in cell-based kind of models, we see that the molecule protects astrocytes, oligodendrocytes, motor neurons against very long chain fatty acid induced cell death. It also recovers mitochondrial function, this is here, reduces the oxidative stress here, and in the case of oligodendrocyte, that I said before, it improves the differentiation and the phagocytosis of myelin debris. Also, it reduces microglia activation, which is also seen in multiple diseases now. When we go to animal models to validate further this concept, we use different models. The first one that we use is a knockout model. The issue of this model is that this is very mild. So you can only see changes at the level of biomarkers, almost nothing at the level of motor function. But it's good enough to look at the pathways, and we see that the same pathways that we see that are modulated in vitro, this is translating in vivo, the inflammation, mitochondrial function, and oxidative stress. So to use another, or to test in a more aggressive model, we use a double knockout model. There's another protein called ABCD2, which is kind of complementary with ABCD1. In mice, when both are knocked out, the phenotype is much more aggressive, and here it can be seen a dysfunction in terms of motor function. When we treat the animals with different doses, we see a nice recovery of motor function with a dose response, so nice kind of data for those that work in vivo. We also look at the inflammatory component of the disease because inflammation, as I said, is very important, particularly in the pediatric form of the disease, but also for the adults there's a underlying inflammation. There's no model for inflammation in CLD, so there's no model of cerebral ALD yet. So we look at other inflammatory models, and in this case a model called EAE, that is a model used for multiple sclerosis typically. And some of the drugs that are today in the market are effective in this animal model. CLD and multiple sclerosis are pretty similar in terms of the lesions that are forming in the brain. In this model, looking at inflammation, we see a very nice reduction in inflammation in vivo at the same dose range as in the motor function. So that's what allows us to conclude, among some of the data that we have, that the molecule may work for both forms of the disease. Just to have some more mechanistic details of what's happening in ALD, for those interested in the basic biology. ALD is pretty complex, as I was saying before, so different pathways that are affected because of this deficiency of ABCD1. The molecule being very selective because it's a PPAR gamma agonism-driven effect, it has multiple consequences. PPAR gamma, once activated, blocks the NF-kappa beta, and this has the anti-inflammatory component. It also modulates different genes that are described to be related with neuronal survival and the oligodendrocyte differentiation. And it also interacts with another protein called PGC1 alpha, which is one of the master regulators of mitochondrial biogenesis, and that is related with all the mitochondrial related effects of the molecule. In the case of the CLD form, there's some other activities that are interesting. CLD, again, is complex in the sense that it's not completely understood what happens there, but it's clear that there are different mechanisms that are involved. To start, there's an increase or a BBB disruption, monocyte activation, the macrophage immigration, microglia gets activated, then there's a neuronal death and the myelination. There are multiple mechanisms occurring there. And we tested the molecule in different settings, looking at these different pathways, and we see that the MIN102 of PPAR gamma agonism prevents the endothelial cell activation. This keeps the BBB intact. Also reduces monocyte activation, reduce the microglia activation in different models. Astrocyte survival is improved, neuronal survival, oligodendrocyte survival. So that's why we think that a molecule that is very specific on the mechanism of action but doing multiple things, we believe that can work in a very complicated disease where multiple things happen. In our opinion, it's very difficult to be efficacious in this very complex disease if you are only targeting one of these different processes that are occurring in the cell, because all of them end up being important at the end. So how to move or how to develop a molecule such as this in a disease such as ALD? Well, when we started in this a few years ago now, the situation was the same situation as many rare diseases, unfortunately, is absence of regulatory guidelines, absence of established trial methodology, no scales, absence of natural history data. So with that, develop a drug in the clinic. So what we did is to follow a stepwise approach that's same as can be followed by other diseases, and first is to try to understand very well which is the efficacy that we need to achieve, or the levels, or the exposure that we need to achieve, or what we need to achieve in animals to be efficacious. Then see how we can study that if it's translating into humans in a first study, in a case of phase 1 kind of a study. Confirm this, and then try to understand the disease progression in the clinic and interact with the key people, PIs, patients, regulators, so on and so forth, to develop or to design the study. So first, to understand how much compound or how much modulation of the receptor we need to achieve. That was where we use all these animal models that I referred to. We developed PK/PD models. That was also allowing us to better understand the general concept of PPAR gamma agonism and show that previous therapies that have been developed with this same mechanism of action were falling by far short of sufficient PPAR gamma modulation in the brain. That was allowing us to estimate the level of PPAR gamma engagement required for efficacy. We went to the clinic in a phase 1 study with healthy volunteers, but also looking at PPAR gamma engagement in the CNS, and also compound levels in the CSF of these patients. And what we saw is that the drug, first of all, was tolerated at the tested doses. Of course, that was the primary objective of the phase 1. But also important, this figure here is very important for us. This is PPAR gamma engagement, and the blue area is the levels that need to be have been seen in efficacious doses in animals. In the multiple ascending dose, we fall just in the right range. So our predictions in terms of the right doses and so on were pretty correct. And this is pioglitazone. This is that drug I referred to at the beginning, a drug that's been used for diabetes. Actually, is still in the market for diabetes. And this is what can achieve. So really falling too short in order to have sufficient PPAR gamma engagement for a CNS efficacy. So that allows us to select the dose for the phase 2/3 trial. The issue with the phase 2/3 trial was that there was no methodology. So it was not possible or not clear, no papers telling us which should be the trial. So we had to define that ourselves. So to do that, we look at the different studies that were around. There was a study where people tested Lorenzo's oil, and maybe you've seen a film called Lorenzo's oil. That's the story of a natural product that was developed in the US and by the same group that identified the gene and identified the disease initially, Uwe Moser in the Kennedy Krieger Institute. The film was done in a moment where people thought that the drug was efficacious, which is not. Afterwards, there was some trials, and it was not showing efficacy. But we had access to that study that included the placebo control group. And we also worked with a group in Netherlands that was now developing or running a natural history study. So we took all this data together, we analyzed the data, and we were able to identify some outcomes that were progression progressing sufficiently in the period of time of that we were planning to use or to treat the patients. So the trial design is really data driven. So it's not what we think that works or that changes, the outcomes that we think that changes, but the outcomes that we have seen that change in a period of 2 years, that is the treatment duration. Afterwards, because this is potentially a pivotal study if it works, we want to validate this with regulators, which is an essential step in this space. So we went to see the EMA and FDA. Of course, we did all this process together with the KOLs of the space and patients. It's also very important for us to have outcomes where patients feel comfortable and so that we can understand the disease from all the different perspectives. So the outcomes, without entering into all the details, we group it in 3 groups, but that's arbitrary classification. What we call more regulatory-related outcomes, which is essentially the primary outcome, which is the 6-minute walk test, together with some other objective measurements, muscle strength, and some scales. But this is also important to get this holistic view of the disease, to combine this with other kind of outcomes, such as those that are more relevant for the patient perspective. And here they are the patient-reported outcomes, which are more and more important in drug development today. Other outcomes, such as the incontinence, this is something that worries a lot patients. And also, of course, the conversion into cerebral ALD. AMN patients can develop cerebral ALD, and this is deadly for them. So we are also looking into that. And, of course, complemented with exploratory biomarkers and imaging technologies that we are using quite a lot. The study was initiated in December 2011. It's fully recruited. Actually, we recruited ahead of time, which is surprising in a way for a rare disease, and even with some more patients than what we were initially targeting. So the team did very well there. And I think that is also showing that there's a big or high medical need in this disease, and also showing that the community is starting to be well organized, because your team can work very well, but these other things are not there. It's usually not that easy to recruit. It's the largest trial ever in this indication, and most of the patients, all of the patients are recruited, but many of them are already beyond 1 year of treatment, and we expect data by the end of next year. So 2 years treatment, no interims, and so on, and you can see all the sites that are involved. So if you are familiar with ALD, you see most of the key people in ALD space in this figure. Then we have CLD. So in CLD, we are not with an active trial there yet, but we are preparing to launch a trial. It offers a unique opportunity, we believe, MIN-102, because we've seen that has different contributions to the CLD progression. So we think that in the early stages of the disease, there's an opportunity for this molecule before patients have to go into a transplant. And we are now in the place of putting together a trial, a phase 2 trial in this case, targeting boys between 2 and 12 years. This case, a trial driven by imaging, brain MRI, and looking into the brain of these patients. And we believe that this is complementary with the transplant. We are not saying that transplant is not going to be needed, but what we know is that these patients progress very rapidly. And sometimes they lose the window of transplant because the donor is not found on time. So whatever opportunity can be developed there that stops or diminishes the disease progression, it's really welcome. And I will finish explaining a little bit what we are doing to expand beyond ALD, and in this case in Friedreich's ataxia, where we did the same process essentially. So I'm not going to be as detailed in this case, but we follow the same approach. First is to try to understand the disease. In this case, is again neurodegenerative condition related with the deficiency of frataxin genes, leading to essentially a mitochondrial dysfunction. So this is the most relevant component of the disease. It has different affects in different ways patients. On one side, there's the ataxia component of the disease, but there's also a cardiomyopathy and diabetes for some patients, not all of them. The onset is between 5 and 18 years in most of the cases, although there are some cases of later onset, and there's no treatment approved again. So again, another disease with a high unmet medical need and very severe condition. So here we did, together with the people that understand the disease and has the animal models, by the way, there are no CROs with models of ALD, nor CROs with models of Friedreich's ataxia. So we need to work with academic groups that know how to run these models and understand the disease. That's what we did in ALD, that's what we did in Friedreich's ataxia. Here we look at different systems. In the dorsal root ganglia neurons, we see an increase of survival and decrease in neurodegeneration. We see an improvement of the mitochondrial function. This is related with this figure here. Upon silencing of frataxin, there's a change in membrane potential. This is related with the function of mitochondria, and this is normalized with the treatment with MIN 102. The restoration of energy production, and also an effect on motor function. So there's different animal models that we tested. So potentially a disease-modifying therapy for Friedreich's ataxia as well. This study started very recently, so we did the press release of the approval of the study a few weeks ago. We have not done the press release of the first patient randomized, but we will do it very soon. Again, as I said before, very important for us to work with the key people in the space. So we are a tiny company. We are not experts in any of these diseases at the end of the day. So we need the experts to work with us, and we are very grateful for that. We did exactly the same process to look at data to select endpoints, not to select endpoints based on what we think that is important, but to look at the data and try to understand. And here we had a close collaboration with a group in Minnesota that is running a natural history study, that they have data that is not yet public, so I cannot show you the data, but that was telling us that there are imaging technologies that can be used to understand disease progression, and that we are using pretty state-of-the-art technology, the imaging technologies for that. Again, it's a shorter treatment, is 1 year, but we are expecting results in parallel with the other study, so end of next year as well. And again, if you are familiar with this space, you see that we are working with the key people in the space as well in the countries where we have active trials, including Barcelona, of course. In this case, a trial is a phase 2 study, so it's more intended to validate the concept than to get a regulatory approval at the end. It's a smaller study, 36 patients to enroll, and mostly driven by imaging. But we also combine this with clinical scales, also some motor tests, biomarkers, and so on and so forth. So just to finish, I have to acknowledge the people that have been helping us to start, those that provided the funding. So we have many VC or venture capital investors from different places, Spanish, French, Italians, Swiss, Belgians. But we also got strong support from different public institutions, local, Spaniards, and also the European level. So we are very grateful for that. And this is a team effort. So this is, as I said before, so this is not something that we can do alone at Minorix with 20 people. So you need much more than that. So that's why we have a broad network of collaborators that they are listed here in this slide. This is more for the non-clinical kind of work. Of course, all the clinical sites are very important for us, and I showed them in the previous slides. Some other people that's been helping us in the process, some other advisors. And last but not least, of course, our team that is working very hard, those that are at the company today, but those that have been with us in the past. And thanks to them is why I can be here explaining this nice story to you. And that's it. And thanks for your attention.
Thank you very much, Mark, for this nice history. It's just a success story, and I hope the best for
if the trial goes well.
If the trial goes well, but I think the preclinical data it's so interesting. I don't know if anyone has any questions. Yeah, I think so.
Thank you very much for a really good talk. I was wondering, the mutations in the ABCD1 gene, are they loss of function or missense? Do you have any residual activity left of the transporter?
There are many different mutations. There are hundreds of mutations. So, can you hear me?
Yes, I hear fine.
The camera.
Yeah.
I need the micro for the video. Yeah. There are many different mutations. So, and we are not looking into that because we are acting downstream of the pathway, and so we don't really care about this point. So, but there are all kinds of mutations, and essentially the disease is driven by a deficiency of function, so lack of function.
Any more questions? Claudine? Wishes to collaborate? Okay, thank you very much again, Mark. We are good in time, so let's move to the next talk. It's going to be by Ivane. She's from this Vall-d'Hebron Institute of Research. She's gonna talk about the targeted nanoliposomes for Fabry disease. So shortly, I will just go with the presentation.
So thank you very much, Mark, for giving me the opportunity to show you here what we are doing in Vall-d'Hebron regarding rare diseases, and specifically trying to find improved treatments for Fabry disease in this case. So I'm very sorry, because initially my talk was going to be before the previous one from Minotics, and it was well planned in that sense, because we are more in the preclinical part of a study. But I was attending a different conference now, so you will need to hear me after listening to these good results in the clinical levels. So just to start, I would like to know which is the background of the audience. How many of, I guess that most of you are working in lysosomal diseases, but how many of you are working diagnostic part of the diseases? None? All of you are working in therapy? How many of you are working with nanotechnology? None of you? Okay, so I will spend some few slides trying to show you what nanotechnology can do for the improvement of the therapy in the lysosomal storage diseases, specifically in those of them that can be treated with the enzyme replacement therapy. ¿Estás?
Yes.
Okay. So the idea here is that I'm coming from a group where we try to use nanotechnology to fill the gap between the basic research and the different clinical needs. We have different research lines. Most of them are based on the treatment of oncology, that, as you know, is one of the main fields in the nanomedicine. But at the very beginning, many years ago, we started working also with lysosomal storage disorders, and there is a reason for that. And the main reason is that most of the nanotechnological platforms, liposomes, polymeric micelles, whatever, they go straight to the lysosome. So we had a vehicle there to work with. So just to start it, as I said, I'm going to give you a brief introduction on nanomedicine, and then I will basically introduce you this European project that is called Smart for Fabry, where we're trying to develop these liposomes for the treatment of Fabry disease. So just to start with, as I said, we are working with nanomedicine, nanoparticles, and the idea here is that we are going to use this nanocyte carrier to act as a driver, as a vehicle for this active principle of cargo. So they can be very diverse in size and also in the constitution, but the important thing here is that these using this nanocyte carrier to deliver the cargo, what we have at the end is a bigger structure than the active principle itself, and this difference in size and in composition is going to bring the nanoparticle and the active principle to a certain site. And not just that, because it's only a way to increase the solubility of the cargo and increase the bioavailability, because the nanoparticle is going to stay much longer in circulation and is going to reach a specific targets, especially if we are decorating the nanoparticle with targeting moieties. And this is going to be the case in the Smart for Fabry project. So this is not science fiction. As you know, many nanoparticles and nanomedicine have been approved. They are already in the market. And here is just to show you that most of them, many of them, are actually liposomes. And if we can have this scheme out of the data from 2016, we can see that 20% of them are actually liposomes. And liposomes are nothing else than a small particle with a shell, a phospholipid shell, and this makes them extremely similar to the plasma membrane that is protecting a drug in the inner part. Usually this drug is an hydrophobic drug, but you can also use this system to vehiculize, to drive drugs that are not that hydrophobic. So there are also two other components that we can have in this structure that is going to make the whole system slightly different. One is the presence of polyethylene glycol, which is going to give some hydrophilicity to the whole system and make them more, let's say, less visible to the immune system. And also the presence of targeting system, as I said, targeting moieties, either antibodies or peptides in the surface that are going to increase the uptake from different cell lines, and also they are going to drive the nanoparticle to precise sites. So let's say that the main nanoparticle known in the field that is already approved in 1995 is this structure, doxil, which is a liposome that is pegylated, and it is also containing the doxorubicin in the inside part. So this has been the first approved nanomedicine in the market, and it has shown us how to drive, how to handle the preclinical part of this type of system that are actually complex, because we are trying to work with different type of components, and it's not that easy from the clinical and preclinical perspective. Okay, so this is what we have. The nanomedicines are already there, especially liposomes, but how can we use them to treat the lysosomal storage disorders? So here we have a scheme of what is being actually used in the treatment of enzyme replacement therapy. And here what we have, just to introduce you the treatment, is the expression of the natural way of synthesis and biogenesis of the lysosomal enzymes, is that they are synthesized in the plasmatic reticulum. They go to the Golgi, where they get glycosylated, and then they are secreted to the extracellular media. So this is just part of the way. They can also go straight to the lysosome, but this is the most important part for us, because knowing that part of the enzyme is already secreted to the extracellular media means that we can also put a recombinant protein in the bloodstream and do the same process. The important thing here also is that this protein has to be properly glycosylated to be able to then internalize again in the cell and go to the lysosome. And this is actually what the enzyme replacement therapy is doing. It's putting the recombinant protein, and this protein can go inside the different cells through these mannose phosphate receptors. And as you can see, the presence of the receptor and the presence of the proper glycosylation is really, really important to have the efficacy of the enzyme replacement therapy. In the case of using nanoliposomes or any type of nanoparticle to deliver the recombinant enzyme, here what we are going to do is that we are going to protect the enzyme from the degradation of the bloodstream, and we are also going to avoid the internalization through the mannose phosphate residues. We are going to use a different type of internalization, and depending on the targeting moiety that we use in the surface, we can increase the uptake for certain cells. And also, we can try to see if we can be able to cross the blood-brain barrier using specific targets. So this was actually the idea of the Smart for Fabric project. We started with a nanoformulation of this type. I will show it to you in a moment. And the idea is that we had some preclinical data supporting that this type of vehicle was going to be useful for improving the enzyme replacement therapy by lowering the clinical doses that could be used in the treatment of the patients, and by also reducing the treatment of the cost, because we were not going to be using a perfect enzyme, let's say. So we were at this stage, and the idea, was to have all the proof concept to support the translation into the clinical trials. And this is the members of the project. There are 10 different partners. Five of them are coming from research institutions, and 5 of them are coming from companies. So it's a pretty big consortium, as you can see here, and it's been tough to start working with it and organize, but at the end we are reaching to a point where we can really speed up all the research that we are doing in the field. So as I said, we're working product is this liposomes. They have 3 different components. They have in the membrane, in the shell, they have DPPC, which is a surfactant, and cholesterol. And the targeting moiety that I was mentioning to you is this cholesterol PEG with the RGD with a peptide at the end. This is inserted in the cell of the liposome and is driving the nanoparticles to those cells expressing alpha beta integrins because this is going to be like the natural ligand of those integrins. With this, the idea is that we can avoid the endocytosis through the mannose 6-phosphate receptors and target these nanoparticles specifically to the endothelial cells. As you know, lysosomal storage disorders are not specific for certain type of cells, but in the case of Fabry disease, there is a link between the presence of the GB3 deposits and the activity on the endothelial cells and the progression of the disease. So that's why we thought of using this RGD moiety in the nanoliposomes. Well, so one of the advantages of using these nanoliposomes and one of the strengths of the project is that to produce this complex system, we can do it in a single step. It's just by using, by putting different components in this pressured system and by using compressed fluids, we can just have in a single step the whole system form here. And this is something not just simple, but also easy to scale up under GMP conditions. And this actually was one of the objectives of the project. So these are the results that we started with, the nanoparticles, the nanoliposomes, when they did not have any targeting moiety and we were using them in vitro, we could see that they were not internalizing the cells, no fluorescent signal. But when we were adding this RGD moiety, the moiety that is going to bind the alpha-beta integrins, the internalization goes up. We have much more nanoparticles inside the cells, and we can also see that by these confocal images here where the targeted nanoliposome has a stronger signal, 30 times more internalization in these endothelial cells. Not just that, but they also go straight to the lysosomes. In this image, we have the lysosomes labeled in green, and you can see that the colocalization of the nanoparticles with the lysosomes really occurs here. But it's this really active. Are these nanoparticles able to bring the enzyme, the defective enzyme, into the lysosomes? And this enzyme that is being delivered is active once encapsulated, and the answer is yes. Here we have the results of the enzymatic activity. When we have the free enzyme, we have this activity around 300 micromoles per hour and milligram of protein. But when we encapsulate the nanoparticle, and this was very strange, we could not explain that very well at the very beginning, we have this increase in the activity of the enzyme. This is probably because, not probably, but we know now that the presence of the lipids there is stabilizing the activity of the enzyme, and that therefore we can have a higher activity. And when we translate that into cell culture system, we can see also that in the presence of the nanoliposomal system, we have a higher reduction of the GB3 deposits. The GB3 deposits are, GB3 is the substrate of the GLA enzyme, the enzyme that is defective in the Fabry disease. And when nanoliposomes are put in culture with these knockout cells with no expression of the GLA, this system is able to reduce these deposits, in a higher extent than the free enzyme. So in this case also, we see that there is an important activity. But what is happening in vivo? And in vivo, we tested the pharmacokinetics of the nanoliposomal system. Here, encapsulating again the GLA enzyme, we could see that there is a higher plasma half-life and also a reduced accumulation in the liver. This was important because, as you may know, the presence of the mannose phosphate receptors is really high, the expression is really high in the liver, and that means that when in normal situation, when you administer the recombinant enzyme to a patient, 60% of this enzyme goes straight to the liver, and there's a poor bioavailability to other tissues in the body. But in this case, we were able to reduce this accumulation in the liver, but have more or less the same presence in the kidney, which is one of the organs that is the target of the Fabry disease, of treatment for the Fabry disease. So at this point, we needed, these were the results that we had when we started the project. At this point, we needed to optimize the nanoparticle, and this is going to, I'm going to show it to you in a moment. We needed also to introduce here a protein that was not going to be a protein that is actually available in the clinical setting. It was going to be a GLA freedom to operate. It means that the whole system would be able to be translated without the sponsor of a big pharma, in this case, because those were the owners of the clinically available GLAs, and they were not participating in the project. We also needed to scale up the synthesis. I will show it to you in a moment. Start with more standardized preclinical testing and complete the regulatory preclinical safety assess. So to start with, as I said, we needed to produce a GLA that was that we could have in enough quantities within the consortium without having the need to buy the commercial enzymes. And we did that. We had high activities in the batches that we were producing, even higher than the commercial one. And now, right now, we are obtaining the same version of the GLA that in the first version was having this semi-his tag just to be able to purify it. And it was active, as you see here. And now we are just obtaining the first batches of the GLA without any semi-his tag in the sequence. So the next step was to introduce the GLA into the liposomes, in liposomes that were improved compared to the first version, because one of the problems that we had in the previous version of the nanoliposomes was that they were not able to encapsulate high quantities of the enzyme. The enzyme was active, but it was we didn't have enough quantities to start with the preclinical in vivo assays, and by adding to the complex this surfactant, the MAKC, we were able to increase the encapsulation efficiency and also the homogeneity of the liposomal system. The second thing was to change the targeting moiety, or at least add new targeting moieties to the nanoliposomes, and this was done by, one of the things was not to change the targeting moiety, but the length of the polyethylene glycol was used to link the RGD moiety to the cholesterol shell of the nanoparticle. And this is important because depending on this length, the nanoparticle is going to be different, and also the hydrophilicity that is going to have around is going to be different. And in this case, we were testing many different lengths, and what we saw is that a specific length of 400 was the best one to have both things, a nanoparticle that was stable and a nanoparticle that could work well in vitro. As I said, we also were testing different targeting moieties, not just the RGD, and these were the ones that we selected to be able to cross the blood-brain barrier. For the moment, there are two of them that are going to be analyzed, the one with the transferrin, and the one with the glutathione. I will show it to you in a second. And also, as I said, the scale-up. in the previous results, we were working with a production that in a batch that where we were obtaining 7 to 10 milliliters in volume. Now we are working with a vessel of 75 milliliters, and they are also now building the whole system and having the first batches with these big vessels with up to 300 milliliters of volume. And this seems easy, but at the end it needs a lot of time and a lot of effort that was done in collaboration with, the work was done with Nanomol in the collaboration of Praxis, which is actually the owner of the patent of this system. The prototype of the pilot line for this big vessel in the GMP conditions is already available and is going to be built in the facilities of Praxis. For the regulatory testings, we are using also knockout models. The knockout model for Fabry mouse, no expression of the alpha-galactosidase A, and we are working in vitro using primary cultures and in vivo using the systems, the mice, just male mice, because as you know, this is an X chromosome-linked disease. And we've been doing different assays. I will show it to you, but before doing the efficacy assess, we started by testing the cytotoxicity and the hemolysis of the new version of the nanoparticles. We didn't see any effect, any cytotoxicity in the tissue cultures. We didn't see either any effect in the plasma coagulation times or in the hemolysis of blood samples. And we also tested that after repeated administration of high doses of the vehicle without any enzyme inside yet, we didn't have any side effects in vivo. So with this, we moved to see if the system was actually working. In vitro, we saw good results because, again, the nanoliposomal system, in this case with the MAKC, high quantities of the GLA, was able to reduce the GB3 deposits in the cells. And in vivo, we are just running the experiments now. Actually, we administered the mice 2 weeks ago, and we are waiting for the GBC results. The good thing is that the free enzyme is working as good as the alpha-galactosidase alpha, and I cannot show you the results right now, but it seems that the nanoliposomes are working even better than the commercial enzyme. We will need some more time to analyze the data. Regarding the preclinical testing of the systems with the peptides targeting the blood-brain barrier, they will be performed by Johanneum University, Johanneum Research in Austria. They have this specific system to test the blood-brain barrier crossing and the pharmacokinetics using this cerebral open flow microperfusion system. And they are actually doing this by using systems that are encapsulating fluorophores, just by measuring the fluorescence emitted by the samples that they are collecting from the brain of the rats in this case. And with that, the idea is that the last two members of the consortium are going to work in the preclinical regulatory assess. The idea here is to test the nanoliposomal system using rodent species, rats, and also beagle dogs. And they are just defining which are going to be the experiments and the assays, the doses specifically that they are going to be using, compared with what has been done already in different type of systems that have been approved for the treatment of Fabry disease. So with this, this comes to the end. This is the timeline that we have. We have completed some of the most of the preclinical assays with the old version of the nano-GLA system. Now we have to complete the testing of the system without MAKC. Initial results are good, but we need to repeat them and do more assays. And if with the blood-brain barrier assays, we see that the targeting system with the transferrin or glutathione is working better than the RGD, we may change the system and introduce those to have the dual targeting with the integrin targeting RGD and the transferrin or the glutathione. But we still need some time on that. The pilot plant, as I told you, is still prepared and being built in the facilities of Praxis, and regulatory testing is meant to start at the end at the beginning of next year, actually. So with that, I hope I convinced you that the nanomedicine will be useful also for lysosomal storage disorders, and that in the Smart for Pharma consortium, we are actually working in trying to see if this system would be useful for Fabry patients. And with that, I'm sorry there was animation in here. I would like to thank all the members of the consortium that were in here, collaborators in here, and funding agencies at the back of this part. I'm sorry because I was using a previous presentation from Orlando. So with that, if you have any question or comment.
Hello.
Thank you very much. I did not get the point how and why you want to reach the mannose-6-phosphate receptor with your liposomes. So
Okay.
when I
So
understood you right, you integrated some integrins or integrin interacting proteins or
No.
what?
We are trying to target the integrins. To be able to target the integrins, we want to take the nanoparticle and drive it to the endothelial cells. So endothelial cells usually they have overexpression, not overexpression, the high expression of certain type of integrins. Integrins are heterodimers of alpha and beta. And there is this sequence of RGD, 3 amino acid, that is prone to bind these integrins, right? So we wanted to do this because we know that the endothelial cells and the stress of the endothelial cells is very important in Fabry pathophysiology. And we thought that maybe targeting and bringing this nanoparticle to endothelial cells could be helpful for Fabry disease. And that's why we were putting the nanoparticle with this RGD moiety. Okay, so this was one part of the issue. The second part is that we were aware that usually recombinant proteins of enzyme replacement therapy, they internalize through these mannose-6-phosphate receptors. And we didn't want to get rid of this type of internalization, but we wanted to have an alternative way of entry. Why? Because if you just rely in the internalization through the mannose-6-phosphate receptor, your enzyme is going to go to those tissues with high expression of this receptor. And other tissues with lower expression are going to have less presence or less quantity of the protein. And by using this alternative system of internalizing in the cells, we could have a better efficacy.
But before you reach the mannose-6-phosphate receptor, you have always to transfer or pass through the endothelial cells, where the mannose-6-phosphate receptor are very low abundant at the apical site. They are more localized at the basolateral site, and you have to transcross the cells before you reach the cells beyond or below the endothelial cells, right? So the liposomes, they give you the perhaps the advantage that they can fuse with the plasma membrane independent of receptors or something, and can be endocytosed with along the endocytic pathway directly to the lysosome. So did you test it whether your your liposomes can be inhibited by mannose-6-phosphate,
Yes.
the uptake?
We didn't do the experiment. We didn't do the experiment. But the thing is that the internalization with the targeted system is much more higher than the internalization without the RGD moiety. So that's why we think that the internalization is specific, because the internalization of the nanoliposome by itself is really low. Right?
Okay,
But it was a good point. I can discuss this with you later. But the thing is that, as I mentioned at the very beginning, this liposomal system is usually used for hydrophobic drugs. So in the case of a protein, we don't have an hydrophobic drug. We have something that is more, in this case, is not really hydrophobic. And what happens is that the GLA, the enzyme in this case, stays inside and also in the membrane. So when it's also in the membrane, it means that maybe some of the mannose-phosphate residues are being exposed to the outside, to the outside of the nanoparticle, to the cells. So we cannot discard that there is an internalization also going through the mannose-phosphate receptors. Because the initial assays that we were performing, they were done in the absence of the protein. So there was no protein in that. We need to repeat those by having the protein present and see if the internalization goes more through the integrins or through the mannose-phosphate receptor.
Perhaps I have also to kill some of your illusions. I have to say that the recombinant enzymes, they do in the maximum contain 20% of mannose-6-phosphate residues per molecule. So every fifth molecule has perhaps a mannose-6-phosphate residue. So this does not play an important role. So during when you injected your liposomes and you said the majority goes to the liver and something. The liver is also not a very high content mannose-6-phosphate receptor organ. So you have an uptake via the mannose receptors and not on the mannose-6-phosphate receptors, only to keep this in mind.
Okay.
But my question is now, how much protein you can deliver in total in your liposomes? You know, the patients need between 0.5 milligram and 1 milligram, sometimes depending on the enzyme, 10 milligram per kilogram body weight. Can you apply so much with your liposomes?
With the previous version of the liposomes, we were able to deliver 1 milligram per kilogram to the animal model. With this newer version of the liposomes, we are able to increase that 5 times. So it's 5 milligrams per kilogram. So still, it's far from what you need, for instance, in Pompe disease, where you need to have 20 milligrams per kilo. But we don't know, because we haven't performed, well, we have performed the assays, but we haven't done the analysis yet. We don't know if your pharmacokinetics is better and you have a longer half-life, you may need to reduce the dose that you are administering to have the same effect. So this is one of our hopes, that we can increase the half-life of the protein in the bloodstream, and with that reduce the dose and the frequency of the administration. But we don't know it yet, yes.
Okay, any more questions?
I have one question about this in vivo data, about the in vivo data in mice models, as you mentioned, that I was wondering, have you already measured, or do you know what is the target bioavailability of the liposomes in the brain? How much of your dose actually comes into this?
So the crossing of this of the liposomes to the blood-brain barrier, we don't know yet how much is going to be crossing. How much will we need to be able to deliver, to cross, to be able to have a reduction in the GB3 deposits and have a clinical effect? It will be probably low, but I cannot give you the exact number. The thing is that in Fabry disease, there are neuropathological disorders, but they are not that they are not the important part of the disease, because the patients are not that much affected. And also, if you take as an example the enzyme replacement therapy, you don't need to replace the whole quantity of the enzyme that a healthy person will have. Just by delivering a small portion of it would be enough.
No,
So
I was just thinking mostly about the nanocarrier,
For the nanocarrier.
the cargo as you are using it. How much of your nanoparticles, 1 million, if you inject 1 million, how many will get into the brain?
I cannot give you the answer. I don't really know. In this case, I don't know. For other type of nanoliposomes, if you are able, that 0.2, 0.3 of the dose, if that is able to cross the blood-brain barrier, they call it a success. So we still need to test it here, so we don't know. We know for other systems that we are using not nanoliposomes, but exosomes, for instance, that with small quantities we can have a reduction of the GB3 deposits. But with this system we haven't tested it yet. I'm sorry.
Okay. Thank you.
Okay, thank you very much. So now it comes the break. We have a coffee break. You will find it upstairs on the right hand. There's a terrace there. So thanks, we have good weather, so we can take advantage of this. So now we will start back at 4. So please, we can go upstairs. Just as precaution, just bring your things with you. Okay, we're at the science park, but we never know. Now it's time from Julian Isla. He's member of the Passion Advocacy of EMA group, and also he's the CSO of a European Federation for Dravet Syndrome. He will join through bio conference, so I'm gonna call him.
Hola. Sí, sí.
Nuestro compartir pantalla, espera, te pongo en pantalla grande. Y ya está. Cuando quieras, podemos empezar.
¿Me podéis oír?
Perfectamente.
Vale, déjame que comparta la pantalla si funciona. ¿Podéis verla? Bueno, pues yo estoy listo. I'm ready. There is some noise maybe coming from your microphone. It's fine. Thank you for the invitation to this conference. My name is Julian Isla. I'm based in Madrid, in Spain. I work for Microsoft as a full-time employee. I work in the Microsoft Consulting Services Department. My expertise, I'm a software engineer. My expertise is right now in artificial intelligence. This is what my group is doing at Microsoft right now. I'm also the chairman and founder of Foundation 29, which is a non-profit organization with focus on rare diseases and how technology can help people with rare diseases to have a better life. I'm also the Chief Scientific Officer of Dravet Syndrome European Federation, which is the federation of patient groups in Europe with people with Dravet syndrome. In Europe, I'm also helping Urordis as a member of the TAG. The TAG is the Therapeutic Advisory Group, and is the group providing advisory services to the board of Urordis. In Europe, I'm also a member of the Orphan Drug Committee from the European Medicine Agency. This committee is very important for rare diseases in the European regulation, because in our committee, we are giving the orphan drug designation to new medical products for rare diseases. And in Spain, I'm also a member of the Scientific Advisory Board of CIBERER. CIBERER is the Spanish network for research on rare diseases. This is my declaration as member of a committee of the European Medicine Agencies. The opinions of these presentations are my own opinions, and I'm not representing the European Medicine Agency on that meeting. The reason I'm involved in so many activities is because of Sergio. I have two kids. One is Sergio, he's the younger one, and Sergio has Dravet syndrome. Something that I remember when I come from the neurologist, the first time I heard about the Dravet syndrome is this word, catastrophic. The thing is that, when you are a parent and you heard from a physician, your son has a catastrophic condition, the things that are coming to your mind are very bad. This is the definition of catastrophe or catastrophic, and really it's a bad situation and generating great problems, troubles, or destruction. This is the phenotype of Dravet syndrome. Unfortunately, Dravet syndrome is a severe neurological condition, and right now we don't have effective drugs. We don't have an effective therapy, The mortality is very high in the younger population. 20% of people die before being adults. From a neurological point of view, you are an adult if you have more than 16 years. They are care dependent for life. On this video, I have a video of a seizure because, you know, for the epilepsy community, seeing or explaining what you have to do when people have a seizure around you is very important, you know, but I guess we don't have time today to go into the video, and we can move forward. This is the video, okay. But the most important thing is you don't have to do anything with the patient, you know. Just most of the seizures, they stop after, you know, 5 minutes or less than 5 minutes. The mother of the child is putting the child, you know, in this safety position. This is the typical position that you have to use when a patient or somebody is having a seizure. And you know, the seizure the kid is starting to shake the arms, and he will start to shake, you know, the legs as well. This is a tonic-clonic seizure, where a convulsion, you know. Okay, the most important thing is you don't have to put anything in the mouth. You don't have to do anything. Just wait for 5 minutes. If this is not stopping in 5 minutes, you have to call the emergency service. In Dravet syndrome, if you are lucky, you have 4 or 5 of these events per month. If you are lucky. If you are not lucky, you can have 20 of these events per day. We have children with a lot of seizures every day. But the thing is, when you have this situation, you have to go to the moon. You have to shoot to the moon if you want to make a change, because you don't have treatment, you know. And this is exactly what I tried to do 9 years ago. Sergio right now is 10 years old. And 9 years ago, I found more patients, more parents with the same problem. I tried to find people in Spain. I just found 6 families in Spain. I had a meeting with them, and I told them, we can go for a cure. You know, we have to go for a cure, because if we are not going for a cure, the cure is not going to come to us. I use the same example that Kennedy was using to the Americans in the 70s or the 60s. We have to go to the moon, you know. It's because it's not easy, it's because it's hard, you know. So it's the same, you know, mission that a lot of parents and families are using right now in the world. But we started to question ourselves if we are patients. You know, patience means waiting. But are you willing to wait when you have a child with 20 seizures per day? I'm afraid we are not. And we started to be impatient. We are not willing to wait. We want to make changes. I founded the Dravet Syndrome Foundation in Spain and also the European Federation in Europe, both, with the help of other parents. And at the beginning, when you are running a patient organization, I didn't have any experience about science or drug development or almost anything, right? So we did a lot of things at the beginning. They are the projects that we did, you know, in both organizations. And we invest a lot of money in research. We invest almost 2 million euros in research, basic science, you know, but nothing was useful after 10 years. Any of the euros we put on those projects has been successful right now. We invest also a lot in technology, you know. You can see right now how patient organizations are investing in technology, devices, software, you know, but we failed as well. Because the thing with technology is that probably you need a business behind you. You need a company, and you need a commercial business in order to be successful. If you invest in technology and you don't have a clear plan of what you are going to do, probably the money you are putting there, this will be lost. Registries is a common thing in Europe, you know. We tried to make the registry for the European population, but we failed as well. Why? Because we run into a lot of discussions, you know, different opinions. Some patient organizations want to put, you know, a registry managed by doctors. Other groups decided to put a registry made or managed by patients. Another registry made by researchers. And at the end, we didn't agree on a common space for the registry, and we failed. The registry is not there. Something that we try to do is phenotype and genotype our population. This is super important, you know, in order to do a stratification of patients. But we fail as well. If we weren't able to make a registry, phenotyping and genotyping was really not going to the moon, but going to Mars. Can you hear me, right? Just to check you are still there. Hello? 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 Okay. Something that we did and worked very well was working on an animal model. When we realized that the animal model for Dravet was, let's say, a model managed by researchers, and they didn't want to share the animal model with other researchers, we decided to finance an animal model from our money and put the animal model available in Jackson Laboratory in the US, and it was very successful because after creating this animal model managed by us, all the different animal models were public in the next months. It was very good. And another thing very interesting that we did is facilitate the diagnosis process. Something that we did is to put a diagnosis program for every child worldwide with potential symptoms of Dravet syndrome, and we facilitate the genetic diagnosis. And we started with 6 families in Spain, and right now we diagnose almost 900 people worldwide, which is a lot. It was very, you know, after 8 years of running these organizations and, you know, a lot of investment, a lot of discussions, the most important thing that we did was invest in animal models, you know, and invest in diagnosis. Orphan drugs, you know, this is a very interesting topic because at the end, if we are talking about rare diseases, we are talking about orphan drugs. And when Sergio is the name of my son, and when Sergio started to have the first symptoms, it was 11 years ago, and the first medication Sergio started to take was this combination of drugs, is valproate acid, clobazam, and stiripentol. Stiripentol is an orphan drug right now. 10 years after, or 11 years after, the medications he is taking are the same. No big changes. The only thing is that we ran a couple of clinical trials in the last 2 years, one for CBD, cannabidiol, and fenfluramine, which are new drugs being developed for Dravet syndrome, but they are symptomatic drugs. They are not drugs willing to solve the problem. These drugs are just treating symptoms. The main thing is that drug development is very, very slow. It's a very slow process. Regarding, you know, medical products and regarding the work that we are doing at DIMA, this is the number of orphan drugs who receive the orphan drug designation in the last years. And you can see very easily the number is going up. But at the same time, the drugs going to the market, the drugs who are getting the orphan drug designations are more or less flat. And the number of new drugs every year for rare diseases is just around 15, which is not really a big number, right? And when you have a drug approved, the company has to go to every country and ask for a price. If the drug is not reimbursed by the national authorities, the drug won't go to the patients. And it's not easy because, you know, there is a lot of economical discussions that the companies has to face with national authorities before being reimbursed. But we have 7,000 rare diseases. The number of, you know, medical products is just 150, roughly, you know. So that means that just this small population, they have access to a drug for the disease they have. The other white boxes are people with rare diseases with any treatment at all. Another thing is that for these drugs, we have repetitive indications. I mean, we have drugs for the same disease. So, and we have a lot of men. So the situation is even worse because we have around 70 drugs for rare diseases, for individual conditions. And it's even worse if you check the efficacy because most of the drugs are symptomatic and they are not being a cure for the disease. The only one drugs that they are a cure is this small box. If you compare this box with the total population, it's pretty much nothing. So the situation is not very good in terms of drug development. And this is the cost right now for orphan drugs worldwide. It's a lot, you know, it's $120 thousand millions dollars. It's a lot, you know. But the thing is, if we want to increase this number to the overall all population, the number will be, because remember this number is just for this teeny box. If we want to increase this cost to all the rare disease population, this will be the number. The number is so high that it's difficult to make, you know, to understand how big the number is. But if I told you, is like the, you know, the general, the gross income from Germany, UK, and France, the overall revenue of these three countries together, you can realize how big it is. Yeah, because cost for rare diseases is a problem. You know, this is the calculation of cost based on the prevalence. The threshold for rare diseases is here in the 10,000 patients. And you can see when the prevalence is reaching this number and the condition is rare, the cost multiplies. Probably in a very artificial way, you know, because it doesn't make sense from a science or drug development point of view that just the prevalence is raising the cost. You know, it doesn't make sense. So in other words, the industry is making money because of rare diseases. Another problem is, you know, how we are repositioning the drugs. I mean, the orphan drugs can be used for other conditions. And as you can see here, and this is the rank of, you know, top rank orphan drugs, the orphan utilization for some of these drugs is marginal. They are developed as orphan drugs, but they are used for other conditions. And this is something that we have to review right now, because it doesn't make sense from a regulatory point of view. Why doing drugs is so difficult? Why I like this sentence from Daria, because Daria says it's because making drugs is difficult because we have to be better than nature. And I agree with that. You know, making good drugs is difficult because you have to change nature at some point, and this is difficult. And the problem is you have to deal with safety, and dealing with safety is expensive and difficult as well. Yeah, I heard some noise. Are you still there? Okay, yeah. I was scared of being alone, talking on my own. So you can follow the presentation, right? Okay. Okay. So the development is very slow. Sorry for this slide in Spanish, and the cost is very high. And on top of this, we have to take care of safety because it's very important. So at some point, I guess we need to, you know, do some kind of disruption to make sure drugs are being developed in a different way. If we are looking into other industries, like, for example, you know, the aeronautical industry, flying is a very complex process right now. You are taking on from one small, you know, space in the ground here in Europe, and you are landing in the US, you know, in some hours with precision, pretty much on time, and safety most of the times. But the thing is, this industry, you know, 110 years ago, doesn't exist. This industry has moved from planes made of paper and wood into these super complex planes, you know, in a very fast way, much more faster than we are doing in medicine. And they are doing this also democratizing the access. This picture is a real picture from SAS, from the Scandinavian Airlines. And yes, they are serving very good food, you know. 30 years ago, 40 years ago, flying was for rich people. But right now, everybody's flying. Everybody's flying with the lower rates of casualties in the history of aviation. This is the number of casualties, of fatalities by year, and it's going down. Every year is going down. More people is flying in safer planes. The aeronautical industry is doing this mainly because this, you know, they call this a black box, but it's not black, it's orange, you know, when they are recording almost everything. Every flight, every commercial flight has been recorded, the conversation of the pilots is recorded, and the industry can analyze what's going on. This is the implementation of the feedback loop, something that when you are an engineer, you have in your brain. In the universities, you are trained when you are an engineer to implement that process. You have to create a system, measure the system, and feedback the full process. But in medicine, we are not working on that way. If I go to my doctor, to my primary care physician, and my primary care physician is giving me an antibiotic, for example, if I die, my doctor is never aware of. Never. It's quite crazy, right? So what we can do? You know, Airbnb is one of the most important companies in the world for hospitality, and they don't have hotels. And Uber, well, you know Uber, they don't have cars, they don't have taxis. What they have, the only thing they have is data. Data about people, data about the business, and the data is everything what they have. And data in the new world is very important because it's the fuel of systems. And it's the fuel of artificial intelligence, for sure. And the artificial intelligence is here to change the world, the way we are doing business, but also the way we are doing science, you know. So my point is the most important thing to take care of from a patient point of view is the data. Because the data is something that we have as a patients, nobody has. And I will give you an example of what happened with the European Medicine Agency 3 years ago, the Diabetes European Federation, you know, because it's a very good example. When, you know, the European Medicine Agency invited me to go to the EMI at the beginning as an observer. The first day I went to the EMI in London. Right now it's in Amsterdam, but you know, 3 years ago it was in London. You are going to Canary Wharf. Canary Wharf is a very, you know, impressive place in London because this is the financial district, is where all the banks, all the, you know, the financial industry is. And in the middle of this place, this is the European Medicine Agency building. So you are going to this committee. This is my committee right now. And they were discussing this application for Dravet syndrome. The application was for the design of the clinical trial. And this lady, Kerstin, the lady in the right, which was the coordinator of the product, asked me to participate on this process. It was my first meeting. I was very impressed by the ask. But the thing is that they were working on protocol assistant. Protocol assistant is something that the companies can use to ask for recommendations for the clinical trial. At this stage, the company working in a product for Dravet syndrome was asking a question to scientific assistant, asking for the design of the clinical trial. And you know, the clinical trials are very important because the inclusion-exclusion criteria are mainly the only opportunity for patients to be access to new drugs. You know, you are designing the clinical trial, and inclusion criteria says the patients going into the clinical trials and the patients going to be excluded of the clinical trial. During this meeting, the discussion was about this product. This product was the only one approved for Dravet syndrome. It's not very effective, but it's the product that we have, you know. And the position of the company was in the design of the clinical trial that they didn't want to include that product in the clinical study. They want to remove or eliminate patients taking this drug from the design of the clinical trial because the position was that just 5% of the European population was using the drug. I went to the meeting and the committee asked me, Julian, it's your first meeting, thank you for coming. How many people in Europe is using Diacomit, is using this drug? Do you know the number? And I say, no, sorry, I don't have this number. Can you imagine in my position, you know, you are representing your community, you're going to the European Medicine Agency. The European Medicine Agency is asking a very simple drug. How many people is taking this drug? They are not asking about pharmacokinetics, pharmacodynamics, drug-drug interaction, or something super complex. They are just asking how many people is taking this drug. I didn't have the number, but I told them, okay, can I go back? Can I back in 2 weeks and collect this information? Because the information is in the medical records of our people. We have the information, but the problem is we didn't collect the information in a proper way. So what we did was a SurveyMonkey, survey. Well, SurveyMonkey is a very important tool for science, as probably you know, and we collect data from 274 patients in just 1 week. The quality of the data was so good that several months after we published the data. But the most important thing is the data we collected, almost 50% of the population was using this drug that the company wanted to remove from the clinical trial. And I come with this information to the committee. I inform the committee of this situation, and they asked to the company to change the design of the clinical trial to include patients with or taking stiripentol. This is the new study that they created because of that. They have to keep the initial study, but they created another one, this one, because of this input. And this was very important because at the end, one year ago, one mother from Sweden sent me this chart. And this chart is the number of seizures his son had in the last months. And you know, this is when his son started the clinical trial. So you can see here how effective it was, you know. And at the end, this is very important because I realized on this chart the importance of the work that patients can do. Without my participation into that meeting and without these numbers, this will be different. So what we are doing in Foundation 29, because of that need, you know, I decided to step back from my roles in the Spanish organization and also European organization, and I founded Foundation 29. Our goal is to help patients to go into this process. This is the cockpit of a Lockheed Constellation, the first plane crossing the ocean from Europe to America for a commercial flight, and they need 5 pilots, 5 at the same time in the cockpit, because you can see it's very complex. It's plenty of things, you know, something that the pilots has to press, check, and use every flight all the time. Right now, this is the cockpit of a new Airbus. Where is the complexity right now? Is in the computers. The computers are doing, you know, most of the work. Yeah, some people can say, yeah, but look at Boeing with this problem with the new planes. But the thing is that Boeing reacted very fast. They identified the problem, and all the planes are in the ground right now. You know, in medicine, you know, you can have a problem for so many years that you don't realize until the problem is so big. You know what I mean? So at the end, what we are looking for is to, you know, empower physicians. The physicians, you know, from our point of view, they need better tools to make better medicine, and this is something that we are doing right now in Foundation 29. One of the main projects that we are running right now is a project for, you know, support physicians with the process of diagnosis for rare diseases, and we use artificial intelligence to support physicians through this process. That's the end of my presentation. This is a real picture of my son in an emergency room in a hospital, and it's just to say thank you to the, you know, to the doctors, the nurses, the people working in hospitals, because, you know, they have to work most of the time 24 hours in a row, and at the end they are taking care of our patients, and this is my small recognition to these people. And also I really like this sentence of Newton, which is very popular in science, and is, you know, standing up on the shoulders of giants. You know, the people is just able to do good, you know, important things, because at some point you need to identify and you know, a giant, and if the giant is willing to put you in your shoulders, you will see, you know, in a long distance and do things better. And that's all. Thank you very much for your time. If you have any questions, I guess we still have 7 minutes.
If you have any questions, I can address. Yes.
Yeah, well, one, it's super complex, you know. If you are working in rare diseases in Europe, or not just Europe, in the US as well, there are just a few registries with good quality, you know. And the problem is who is managing the registries? Because in the traditional world, the registry is a database that has to be managed by an owner. And the thing is, in the past, this kind of databases, they have been managed by physicians or researchers, right? This is okay, but the problem is that the data is changing, you know, and it's very difficult to keep these registries updated, because if you don't have the patient involved, the data will be obsolete and old in a few days, in a few weeks, you know. So this is the big question, and I'm really pushing to, you know, give or empower patient organizations to take ownership of this. But the problem is most of the patient organizations, they don't have the knowledge or the infrastructure to maintain a registry, you know. For me, this is the main problem right now, is who is managing this registry? Because I don't think, you know, the physicians can manage. I don't think the researchers can manage. And other things like privacy, security, are preventing the use of data or the use of registries will be massive, right? This is an area where we are working in Foundation 29. We are providing registry solutions to patient organizations for free that they can use in order to collect the data and make sure the data can be useful for researchers, industry, for drug development, and keep the data safe, right?
So any more questions from the audience? Yeah. What's your name? Your José Maria from the audience is just first of all just to thank you for this presentation, and he's asking about what kind of tools are you using for clinicians. Sorry.
Tools that we are using for clinicians, or tools that we provide to clinicians?
That you provide to the clinicians, what kind of tools and presumably what kind of functions they are just enabling them.
Yeah, the most important, right now, the only one product that we are offering to physicians is that product. I guess you can see my desktop, right? and we call this DX29, which is a supporting tool for diagnosis, you know. And with this tool, you know, the physician can do automatic phenotyping. I mean, you can upload your medical record or the medical records of the patient, and the system is extracting the symptoms automatically. We are doing genotyping in terms of we are analyzing genomes and exomes and prioritizing genes based on the phenotype that the physician is putting into the system, and we guide the physician through symptoms identification. There's a system that when you are putting this information, the phenotype and the genotype, the physician is receiving recommendations for new symptoms that you can select if the patient has those symptoms or not. And at the end, we are, you know, building a set of conditions matching with the phenotype and the genotype that you can use to do your, you know, differential diagnosis. Right now, this is working in a beta stage. Okay, if you are a physician, you can sign up here and test it. You know, everybody can do it, and we are using, you know, software from universities and research centers worldwide to make that possible. Yeah, all the information is here. Okay, this is just one example, and this is the most important product that we are working right now.
There's some time for one last question.
Yep.
Yes. There was one question regarding which are your thoughts about the ways, the strategies to lower the price of the orphan drugs.
It's a good question. Probably we will need another hour to go deeper into this question.
Yes,
for me, you know, the most important thing will be to collect data and make sure the data are reusable, available, and reachable. Because the problem is when you are developing a new drug, you know, the risk that you have to take is very high because the unknown piece that you don't know, you know, the companies, the industry, they have to make a big risk when they are, you know, developing a drug. And why they are making a big risk is because they don't know a lot of things about the drug. They don't know if the drug is going to work. They don't know about the specific population more, you know, more, let's say, with more chances that the drug will work. The problem is also when if the industry is making a mistake and they have an issue with the drug and a patient die in a clinical trial, the product will be killed automatically, you know. So let's say the industry has high, you know, reaction to risk, and they are not willing to take risk, and this increase the cost. And also there is an artificial cost because of how the drugs are developed. You know, the industry, they have stakeholders, they have to look for revenue. This is a business, and they are designed to make money. So at the same point, we are paying the money that the stakeholders are receiving. So I'm really questioning myself if the, you know, the business model behind drug development will be valid in the next years, you know, because right now we are in a situation that the data is coming from the patients. The patients are producing the product. The product is being developed by a company looking for revenue, for commercial revenue. The company will ask for a price to the government of the patient. The government will have to pay a lot. The money is coming from the patient. So it doesn't make sense, you know? And at the probably, guys, you know, because you are researchers, the work that you are doing right now is going to be taken by a commercial industry. And who is going to receive the revenue of that process? You, the patient, or mostly the company? Is that fair? Sorry, but you know, I answer with a lot of questions, but I don't think the model that we use right now will be valid, you know, for 7,000 conditions.
Yeah. Absolutely. Okay, so I'm gonna finish this talk. Thank you, Julian, for joining this workshop, and say goodbye.
Thank you, guys. Enjoy the meeting. Have a nice conference.
Thank you.
Thank you for the invitation.
Bye. So, let's move to the next talk. It's by Mireia del Toro from... The next talk from Mireia del Toro from... No, the Vall d'Hebron Hospital. And, okay, this is you can control.
Okay. Good afternoon. The next talk is something different. We are going to have a review of all the different approaches that we are using in clinical trials, in clinical practice for the treatment of some of the neurolysosomal, the most frequent neurolysosomal disorders. Esto no funciona. Sorry. Si no me pongo ahí voy pasando. No, es igual con esto. Okay. Well, this is my disclosures. This is a hospital where I work. It's the biggest public hospital in Catalonia. It has pediatric population and adult population. It's a university hospital, and it has a big research institute where Ivane is working, as you had heard before. And as other centers in Spain, we are a reference center for metabolic disorders in a national and a European level. And in lysosomal disorders, we have a big clinic, mostly because we have a lot of adult patients with the most frequent lysosomal disorders that are Gaucher-Fabry. And that's why in the last years we've been focusing in the treatment of this group of patients. Now, this is what happens. I'm sure that some of you are already familiar with this slide. This is what happens in a physiopathological way when you have a lysosomal disorders, and this is the different approaches that from the therapy we can have. The first approach will be the gene therapy to correct the gene mutation. There's a mutant protein that's normally misfolded, and there are some treatments that will try to improve the foldment of the protein and give an activity again. Then there's the deficiency of the enzymes inside the lysosome, and here we have the different enzyme therapies. The deficient of the activity of the enzyme inside the lysosomes will lead to an accumulation of the substrate, and here we have the different types of approaches for enhancing the exocytosis and also for the inhibition the substrate of this storage. And finally, but not less important, all the second abnormalities that we already have heard about and that we know in the last years that are mostly as important, at least, as the substrate storage. And in some of these in some of these groups of therapies, we have an approach for neurolysosomal, but as you'll see, not in all of them. Now, the biggest problem with neurolysosomal disorders is the blood-brain barrier. The blood-brain barrier is a good friend when you have a healthy brain, but it's a bad enemy when you have a brain disease because it doesn't help the different products to get inside. But also, as we've seen before, it's not an absolutely closed barrier. There are different ways of crossing the blood-brain barrier, and there are a lot of therapies that are trying to find these physiological ways to put the different pharmacological therapies inside the brain. And some of them we've heard before with nanoparticles, but there are also some others that are with binding the enzymes to, or the proteins to transporters or to antibodies and to different proteins. In most of the trials we've had since now, the way of putting the drug is by direct injection, and I will talk about this later. But as I told you, there's also some trials going with modifying proteins and transporters. The problem with modifying proteins in animal models is that they've shown that there's a concentration of the drug inside the blood-brain barrier, but then it's not so good the concentration inside the neuron. So it seems that the drug is able to cross the blood-brain barrier, but there's a lot of concentration in the intercellular space, but not enough concentration of the drugs inside the neurons. That doesn't happen with a direct injection. I will talk about gene therapy. We've heard there are a lot of things ongoing in gene therapy for different neurosomal disorders, especially for MPS 3. I think it's well, do you hear me? It's enough? Okay. There's a lot of things ongoing, especially for the most neurodegenerative disorders, as MPS 3A, that's Sanfilippo syndrome, for MPS 2 neurological, the neurological group, and also for MLD. That these ones are the ones that have already results. And as we saw before, there's also a lot ongoing for the group of the lipofunctionosis. Some are already in clinical trials, and some are in preclinical stages. I will not talk about chaperones. There's only one chaperone that's now beginning to be heard in the neurolysosomal group of disorders, and this is ambroxol. But there's not any clinical trial ongoing, so there are only some published cases, and so the results are not positive for the moment. And I'll focus more in enzyme therapies. That's what we have more experience, especially in the pediatric group. You can improve enzyme concentration inside the brain by two ways, or inside the cells. One is with the transplant, hematopoietic cell transplant. That's a physiological way of giving enzyme, and it's the indication for some MPSs. And the other is with the enzyme replacement therapy. That, as you may know, we have available enzyme replacement therapy for a lot of lysosomal disorders. The problem with this enzyme replacement therapy is that it's intravenous, and of course it doesn't cross the blood-brain barrier. I will not explain this because Ivane already explained it before, how the enzymes go inside the cell mediated by endocytosis and the mannose 6-phosphate receptor. But the problem with the neurological lysosomal disorders is it doesn't cross the blood-brain barrier. So what we are using for enzyme replacement therapy is basically the direct injection into the spinal fluid. And what is seen, or what we have seen, it's in animal models, not in our research. What is being seen in animal models, that it's the same if you introduce the lysosomal enzyme in the intraventricular space or in the intrathecal space at the level of a lumbar device. And the absorption by the brain seems to be very similar, and the effect should be quite similar. It's more than 10 years now that the first studies began with intrathecal MPS and intrathecal enzyme replacement therapy, and the last with small animals, and then with big animals, and then a lot of different trials began focusing on the neurological, especially on the neurological MPSs. For MPS 2, that's one of the first of the ones that have MPS and an intrathecal enzyme treatment. The first part of the trial showed that the enzyme was well shown in the CSF. And there was a good decline in heparan sulfate, that's the more neurotoxic metabolite in the MPSs. But then the clinical results in the phase 2-3 of this trial didn't have so good results. And now at the end, it has shown that there's a very small group of patients that are responding to the drug, but not the global population of the patients studied. So the FDA has permitted the trial to go for 1 or 2 years more to see if they really can detect in a small group of patients that the therapy has been effective. For MPS IIIA, the situation was similar. We participated in this trial, and the problem is that here it was before the other one, and here the FDA didn't give the authorization for prolonging the therapy more than 1 year. This was a trial randomized in 3 arms, one was with placebo, and the two others were with different dosing of the enzyme in a device in the lumbar level. Again, what we saw was that there was a decrease in the CSF heparan levels, but then the cognitive decline didn't stop. It was only a few group of patients that they had a stabilization. This is the placebo group where they all declined their EQ. And then in the treated group, there was some patients that stabilized, but as a group, it didn't work, and so the trial was stopped. They looked for antibodies to see if the antibodies against the drug could have a paper in these bad results, but it seemed that there were high levels of antibodies, but there were no neutralizing. There's another trial ongoing now for MPS IIIB. It's similar to the one we participated, and again, the levels of heparan sulfate lower very quickly. What they think here is that the enzyme given intrathecally is also present in the systemic circulation, and there's the liver volume and the spleen volume of the patients goes down. So we have the enzyme can go out the brain to the peripheral circulation. And again, here there's a cognitive decline has not shown a good result. Some patients are stabilizing, but then some patients are maybe improving a bit, but the global results are not favorable. These are the results with the first 5 patients. Now there are 15 patients on the trial, so we will have new results for the next years. The only intrathecal enzyme replacement therapy that's got done very good results is the intrathecal therapy for CLN2. That's terliponase alpha intraventricular. This is really when the patients are given the enzyme in the first stages of the disorder, it really stops progression, and the results are very, very positive. It's approved already by the FDA and the EMA, but in Spain it's not yet approved. We can give it by compulsive use. Now, when we cannot give the enzyme, or there's not ability to give the enzyme, or the enzyme is in the membrane of the lysosome, so it's not easy to give an external enzyme, we can find other ways of trying to exocytase the storage substrate, and this is especially of interest in Niemann-Pick disorder. It's a disorder where there's an accumulation of cholesterol and of some other lipids. And here we have 2 molecules. One is cyclodextrin. Cyclodextrin is an oligosaccharide which has an hydrophilic exterior and an hydrophobic interior, and it doesn't. We don't know exactly how it works, but it seems that it links to cholesterol, and it helps the cholesterol to go out of the lysosome and out of the cytoplasm into the bloodstream. And in animal models, there was a very good result. So there was a clinical trial. There was a first part of the clinical trial in phase 1-2 that was only for side effects. The only important side effect that was registered was it gives the hypoacusia. It affected the acoustic nerve, and some of the patients lost hearing. But in this trial, they already saw that this is the increase in the Niemann-Pick score. The increase in the Niemann-Pick score gives you an idea of how the patient is worsening. And then between the placebo group and the treated group, they already saw that the treated group were not deteriorating as much as the placebo group. And especially, well, the hearing was worse because it's a neurotoxic substance, but in some of the scores, like speech or cognition, it's not only didn't deteriorate, but it improved. So this moved on to a phase 2/3 prospective study in which we participated in our center with a high dose of cyclodextrin. With a lower dose of cyclodextrin because it was seen that it was enough. And the problem in the results again was that there was not shown a significance with the placebo control group because the placebo control group didn't go worse either. So the placebo control group didn't worsen in the Niemann-Pick score, and then the trial group didn't show much difference. They all went better. So now the FDA has approved the 1 year more of the trial to see if the results are different. Another molecule that acts trying to promote the exocytosis of cholesterol is efavirenz. Efavirenz is a molecule that's used for the treatment of VAIC patients, and it's shown to lower the cholesterol intracellular and intralysosomal, and we are also trying to begin a trial with a group in Madrid that's investigating the molecule. There's no inhibition substrate pharmacs for neurolysosomal disorders, so we move to two new molecules that are trying to, as we said before, there are a lot of secondary cellular abnormalities that are shown to be as important as storage, and these abnormalities are related to deficiencies in trafficking, in signaling, in metabolic stress, in oxidative stress, in mitochondrial dysfunction. So there's a lot of things ongoing, neuroinflammation. There's a lot of things ongoing here that that worsen the prognosis of the cells that have the lysosomal disorders, and they help to the cellular death. And then one of the molecules that is being used for ELA. For ELA is lateral sclerosis amyotrophy, and it's also used being tried now for Niemann-Pick C disorder, and it's going to be now tried for other neurolysosomal disorders. Is arimoclomol. Arimoclomol is a small molecule that's oral, and it goes well at it crosses the blood-brain barrier. And what arimoclomol does is it increases the levels of the heat shock protein 70. Heat shock protein 70 is a molecule, is a factor that it's increased when under cellular stress. And this heat shock protein is responsible for trying to help the cell to improve their status and to decrease the possibility of cell death dysfunction, and arimoclomol increases the levels of these heat shock proteins. And what it was seen in the natural history that was performed before beginning the trial was that the patients with Niemann-Pick C, the levels of heat shock protein were very low compared to the controls, to the healthy controls, and that's why the trial was begun. The trial was a phase 2/3. They didn't require a phase 1/2 because it was already in another trial for ELA, so the first part was already done. They directly passed to the phase 2/3 with 50 patients. That was under two of three were with arimoclomol, and one was with placebo, so 2-1. And they could patients could be under treatment with miglustat. That miglustat is the only approved treatment in Europe for Niemann-Pick C. The study was for 12 months, but again, it's have been prolonged for 2 years now. And the results have been positive. The difference between the patients and the treated patients and the baseline was a decrease in more than 1 point if they were not having miglustat, and if they were under miglustat in 1 year, the decrease of the score was more than 2 points of difference. So it was a quite positive result. There was a big change in heat shock protein 70 levels compared to the controls non-treated, and there was also a big reduce in cholesterol and in his metabolite. And they also did some studies on oxidative stress, and they also demonstrated that the oxidative stress of the cells was reduced. And the last molecule I wanted to talk about is trehalose. Trehalose is also another molecule that's now beginning to be heard, and it's beginning to be heard for treating neurolysosomal disorders. Trehalose actuates improving the concentration of TFB. TFB is a molecule that under normal conditions of non-stress and anabolism is inactivated, is in a phosphorylated stage and is inactivated, while mTORC is anchored to the lysosome. When there's a catabolic stress or there's an oxidative stress or any stress to the cell, like a lysosomal disorder, then mTORC separates from the lysosome. There's a synthesis of calcium, and then this activates calcineurin. Calcineurin, the phosphorylates TFB, and TFB goes to the nucleus. That's where he acts normally, and then there it improves. It enhances the autophagy, the lysosomal expression of different genes, and these genes improve they increase the synthesis of lysosomes. They decrease, they upregulate some lysosome hydrolase, and they improve quite the synthesis of new lysosomes with healthy enzymes inside. And they also have a paper in autophagic mechanisms. There have been preclinical studies in MPS IIIB mice. With these mice, they have shown under oral treatment with trehalose, intravenous treatment with trehalose, to live longer. They have they have clinical improvement, and there's also a clearance in the autophagic vacuoles and in the accumulation of heparan sulfate in neuronal and glial cells. And there's also activation of all the different markers of TFB transcriptional network. And we are now trying to begin a trial for MPS III patients, but it's a molecule that will probably be heard for other neurolysosomal disorders also, because it's not specific for MPS III patients. And then my last slide is to show how we can collaborate with the different research groups. Now we have a lysosomal disorders group in the Spanish Pediatric Neurologic Society, also in the Spanish Inborreros and Metabolism Society. We will try to fuse both of them and to do one only lysosomal group. And here we are mainly pediatricians, but we will try also to incorporate the adult physicians that are more involved every day with neurolysosomal disorders. And we have it's not a registry because it's not, it's only a database of the neurodegenerative lysosomal diseases in Spain that is going to be, it's being actualized every year. So we will have at least we will know how many patients we have in Spain and who are the doctors. The only thing we know is how many patients, who are the doctors that take care of these patients. We don't have any personal data or clinical data from the patients. But this is something we are actualizing every year. So it's another thing we can share with you. And that's all. Thank you.
So now, if there is any question or comment, no, just one left, so we can just move on. Next speaker is Alfonso de Olayarzabal from the San Juan de Deu Children's Hospital. He's going to talk about synaptic metabolism. If I'm not wrong, this is this one. So.
Okay, thank you. Okay, so I'm going to talk about the study of Rett syndrome in terms of synaptic metabolism and how we are trying to search for new therapeutic options based on this concept. So, well, first off, yeah, it's not working. Mireia, esto no va, ¿no? Ah, vale. Okay. Okay. So, first of all, we're talking what's synaptic metabolism. Okay, so synapses, we all know, is like the way of communication one neuron to the other. It's a process, like synapses is a process by which two neurons communicate. And what we define, like synapses are really complex processes. in which multiple reactions take place, including all the basically recycled systems, all the receptors docking and production, all the energy needed for the synapse to work out. So all those biochemical reactions that make the synapse to be a synapse are what we call synaptic metabolism. And we are trying to study diseases from that perspective, like trying to study what's going on with the synaptic metabolism with all those biochemical reactions to understand the disease instead of going to the symptoms to the disease. And based on that, we also are studying, among those diseases, Rett syndrome. What's Rett syndrome? Rett syndrome is a neurodevelopmental disease in which many events take place, including seizures, loss of speech, and purposeful use of hands, apnea, stereotypes. It's mainly a neurological disease. This is children, mostly females, are born normal, are born like phenotypically normal. They have a normal early development, and at the age of 6 months, they start experiencing a regression on their development and experience all those symptoms. And what's happening? Like, why is it mainly neurological disease? This is because most of the cases are due to a mutation in MYC-P2. MYC-P2 is a X-located gene, and it's in the X chromosome, and it functions as a master regulator. It binds to other genes, and sometimes it inhibits the expression, and in some other genes, it enhances its expression. MYC-P2 happens to be involved in many pathways, both regulatory, of expression, the acetylation. Is like, those are some of the pathways that it's involved in. So when you touch MYC-P2, you touch everything in the cell. And this is what's happening. Like, to approach the disease, we need to break it down. It's such a complex disease, you need to break it down into events to try to study that. So pretty much, we could divide the neurological dysfunction in this case in 4 terms. First of all, there's an imbalance in the neurotransmission. There are some neuroinflammation processes. There's a defect on the bioenergetics, and there's a problem with neural maturation, among many others. But those could be like 4 pillars of the disease to target. So starting for the first one, we're trying to address all of them to treat the disease or to find new treatment options. So regarding the neurotransmission imbalance, all the neurotransmitter systems are unbalanced in these kids. They have everything to be done, but the main attention should be put towards the glutamate and GABA neurotransmission, as has been shown to be responsible for the main characteristic of the disease. So regarding that, glutamate is the activator, the synaptic activator. GABA would be the silencer, the silencing neurotransmitter. So they will, in a fine tune, they will both equilibrate the communication, saying go on, go off, and in this case it's unbalanced in terms that GABAergic neurotransmission is not properly working, and glutamate neurotransmission is enhanced. So they are like hyper enhanced. So regarding GABAergic neurotransmission is quite a complex system as all of them, and we started to put our attention towards the ionotropic receptors. Well, we know just going fast that receptors can be metabotropic or ionotropic. Those account for the slow neurotransmission, those for the fast hyperpolarized neurotransmission. And GABAergic ionotropic receptors are a huge family of receptors with many subunits forming pentameric receptors. Those are the target for many known drugs. And why we did focus on those? Well, they inhibit the action potential. And why did we focus on that? Well, because GABAergic neurotransmission, to be an inhibiting neurotransmission, it depends on the chloride concentration in the neuron. And it has been known that during development, there are 2 channels, KCC1 and KCC2, that switch the production, the expression, to make GABAergic neurotransmission from positive activator to inhibitor. And it had been shown by our group quite a few years ago that this switch from activate neurotransmission to inhibit neurotransmission is not happening in MYC-P2 patients. So we started to accumulate evidence that there was something that we could do there. And also because all the scientific community is pointing towards this neurotransmission as a big pillar of the disease, meaning it was described how the GABA dysfunction signaling mediated some stereotypes in Rett syndrome phenotypes. And also more important, how MYC-P2 restoration, expression restoration in GABAergic neurons was sufficient to rescue multiple of the in mouse models of Rett syndrome. This is really important because this says that GABAergic is important, and also if you restore GABAergic neurotransmission, you can you can target the disease. So that's pretty big. Well, also there were like many evidence saying like how GABAergic neurotransmission was affected, but yet all of them, like many of the conclusions were like it's somehow affected, but we don't really know how and when it's affected. So our two main questions at this point were, well, how is MICP2 function affecting the GABAergic receptors themselves? And second, can we actually, based on that, define a new therapeutic target? So we did set for this in collaboration with the EDBL and Neurodevelopment Lab at the University of Barcelona. We did set a model of 4 different scenarios of growing complexity. This is, we started on simple cells, then we moved to neurons, to the animal models, and to brain samples. So starting single cells, what we did was a really simple experiment in which N2A cells, which are like transformed immortalized culture cells, we first of all inhibited or blocked the expression of the endogenous MICP2, and then we re-express either MICP2 in the human form wild type or mutated. So first of all, we did see that when we inhibited MICP2 expression, we did see a decrease in the expression, and when we re-expressed the human wild type form, we did see an increase in the wild type, which will not happen when we express the mutant form. So this pretty much said, okay, we have a valid system of inhibiting and re-expression. So with this system, let's see what's happening in the receptors. We took one subunit, GABA1 receptor, which is the main subunit of these ionotropic receptors, and we did see that when we overexpress the wild-type form of MICP2, we accounted for a huge expression of this subunit. This is MICP2 is actually working as a positive regulator of GABA1, which is this GABA receptor subunit. Then we next switch to a more complex model. This is culture primary neurons. Well, they're not well seen because of the light, but what we did is the same. Let's inhibit the expression and re-express in the wild type or mutant form. And we did see, first of all, that we could do that, like re-express MICP2. And that when we did that and targeted GABA1, this is GABA in green and in red and GFP in green, only when transfected with the wild-type form, we will see the re-expression. This is, when we express a mutant form of MICP2 in a culture neuron, we didn't get to express this subunit, this receptor subunit. This is, again, MICP2 works as a positive regulator of GABA1 subunit. Yeah, this is all the same, but just to be published. So with this, we concluded that there is a straightforward relationship between MICP2 and GABA1 receptor. But what happens here is that we will be working in a very, very simple model. And we have to take account that Rett syndrome is a neurodevelopmental disease. This is, kids are born normal. When they are 6 months age, they start doing the regression. So we cannot focus on just culture models. To get that, what we did was work with the mice to see how was this affecting during evolution, and with that, like, increase the translational value of the study. We work with female mice at 2 times. P33, this is 33 1-month-old mice, which are female mice, which are asymptomatic, and 6-month mice, which are already symptomatic. And we targeted MICP2 expression and GABA1 receptor. And we did see a decrease on GABA1 receptor expression at 1 month, which was not accounted when the mice were symptomatic. This is really important because so far studies have been done with the symptomatic mice, which is what you want to treat. And what we are seeing here is that some of the main alterations are actually in pre-symptomatic individuals. And also that these results, the results we are observing, are temporally dynamic. So at this point, we know there is a straightforward relationship between MICP2 and GABA receptor. Then we have dynamic results and pre-symptomatic damage. We say, okay, let's see how is the GABAergic pathway in the patients. As for that, we perform a few experiments of RNA-seq. This is, you take all the RNA in a sample, put it to cDNA, and measure the expression of every gene you want to measure. we did that with the brain of 2 patients who had died at 15 years old at the hospital. So we had the brain sample at the hospital, and we analyzed these brains. The only problem, one of the problems, we didn't have a control of a healthy brain, because you don't have healthy brains. So what we did was compare the results first with an inter-experiment with another patient for an experiment control, and then compare the results to all the publicly available results. So this is, you cannot say with that like how many times it's incremented or decreases, because well, you don't have that such exact data, but you can say the tendency, how fashion it's at. And what we saw, it was in those patients an tendency for overexpression of the result. At first we were surprised for that, because we had seen first a decreased expression. But when we thought of that, well, we analyzed the rest of the pathway, like the whole pathway, and we, overall, we actually saw an or non-modified or overexpression of most GABAergic genes. So we were a bit surprised by that, but when we thought of that, what we had seen was temporally dynamic disease. We were seeing patients at the end of the disease, like who had already died. So yeah, that's not that surprising if you think of that. Like it corroborates our results that when you are pre-symptomatic, you might have something which has seen in mature models, but when you actually grow through the disease, you get another symptoms or another phenotypical symptoms. So pretty much, MICP2 acts as a regulator of GABA1 receptor. There is a huge importance of the time frame, and GABA1 could be a therapeutical target with deep need of putting the attention towards before the disease, like early diagnosis. Well, this is to be published. This is under revision now, and actually this study has been funded by Mi Princesa Red, which is a patient association. So it actually increases like the value for us, because it's for these patients straightforward. So we say that there was this neurotransmission imbalance, and also neuroinflammation, bioenergetics, and neuronal maturation. So regarding all the other ones, we're actually working with minorics and in collaboration with some other groups too, with Min 102, which actually just has been exposed before. So I'm not going through how this works, because we have already been told. We know it's PPAR-gamma agonist. And what we want to do here is to counterpart the oxidative stress, increase the mitochondrial biogenesis, and decrease the inflammation. So, well, first of that, to know that, we know through the literature that this might be affected in Rett syndrome, but we need first to set the ground base to our disease or to our models. So again, we have set up a scenario with increasing complexity models. First of all, in cells, this time it would be fibroblasts of the patients. Then we move towards neurons of the patients. This is through iPS cells differentiated into neurons. And finally, to the mice model of Rett. And we are first going to look for the mitochondria, and then for the neuroinflammation and whole brain affectation. So regarding the mitochondria, we have set 5 parameters, which will be the oxygen consumption of the mitochondria, the ATP production, the reactive oxygen species generation, visually shape the shape of the mitochondria through electron microscopy, and the dynamics of the mitochondrial network through different dynamical markers. So regarding the respiration, we have done a few experiments of a technology called Seahorse, which is like plate respirometry. You measure the oxygen consumption in response to different drugs, namely oligocin, FCCP, and antimycin A-rotenone. And yet these are preliminary results, but we did see a decrease in the oxygen consumption. As for these are extremely preliminary, so shouldn't be taken into huge consideration. But we do see definitely some alteration at the oxygen consumption. Yet real preliminary this. And these go together with the ATP consumption. This is, okay, your mitochondria consuming less oxygen, but what's happening with the ATP, which is like the energetic function. And for this, we actually can rely on this. We did see a huge decrease up to almost 50% on the mitochondrial ATP between Rett patients and controls, Rett fibroblasts and controls. This is we do have fibroblasts, or due to MYC-P2 mutations, we do have a decrease in the ATP production in these cells, like great decrease. This is good as, well, it is bad, but this is good in terms that we can target that to try to solve something. We next move to the reactive to oxygen production. We studied first the anion superoxide anion with the probe mitosocs, and we saw an increase of like almost two-fold increase of ROS in the patients compared to the controls. When we analyze the detoxifying enzymes, this is superoxide dismutase and glutathione peroxidase, which would take care of the reactive oxygen species and release them from the cell. We saw no reliable differences between those two enzymes and patients and controls. And then this is quite a shame. ¿Puedo apagar la luz ahí quizá? Well, we next move to analyze with MitoTracker. This is a mitochondrial probe that marks the whole mitochondria network. And well, I'm not sure if it's going to be seen. Okay, so in controls we did see, well, it's not really well seen, but you have to trust me. We did see the shape in tubular mitochondria, like, you know, the typical mitochondrial network you associate with healthy control, while in red patients we saw mitochondria in donut-shaped mitochondria, which are usually associated to stress and energetic deficits and poor docking on the receptors. So it did fit in terms that yet we have to consider that these are fibroblasts and not the actual neurons. But if in the fibroblast you are seeing these, and fibroblasts are not a type of cell that relies on mitochondria much, what's going to be happening in neurons? If it's the same, donut-shaped mitochondria are associated with poor docking of the receptors and inactive synapses. So it makes sense. It actually fits with the expected results. We analyzed some markers associated with fusion and fission events. As for fission, we had DRP1 and FIS1, and we did see a decrease in the expression. Well, not for this patient, but for these two, and in FIS1. Decrease in fusion, and we studied mitofusin 2, which is a fusion marker, and we see an increase in fusion. So, yeah, sorry. Pretty much summing up this part, we did see a decrease in ATP synthesis, a decrease in oxygen consumption, an increase in superoxide and in ROS production, and an altered dynamic in donut-shaped mitochondria and increased fission. So next would be analyzing all these events in the treatment with MIN 102. When we have that, we will move toward more complex systems, which will be iPS cells, and then to the mice models. Well, I'm the one who have explained this, but that's not me who has been doing that. Well, I have doing that, but I'm part of a big team in which we have like clinical people with Mar and Ocala and Dr. Ocala here and Merce Pineda also. We have the genetic team with Dr. Judith Armstrong. We have neuropsychology team also, and the molecular lab. We have a huge team, and we are led by Dr. Ángel García Cazorla. Like, the brain behind all this is that. And we are funded by, well, mainly funded by different agencies, and also usually collaborate with several groups. So that's it.
Okay, thank you very much for this nice talk. Let's try if we can switch on the lights. Otherwise, so if there are any question about Alfonso talk, now it's the time. Otherwise, we can just finish this workshop. Just to say thank you to everyone who has attended, especially for the speakers, and I think we have a very quick overview about the rare diseases, some of the rare diseases, and some insight from different ways. This is the the idea behind, to bring together people from the clinics, people from the research and discovery and targeting, and also from the patient perspective. I do hope that you have enjoyed somehow. I also wish that you have some new ideas, fresh ideas with other partners to collaborate in the short future. And just to close down the workshops, saying thanks for coming again, and this is it. Thank you very much. And finally, for those who we're meeting tomorrow, just to remember that today we have the consortium dinner. I think you have all the details to reach. If you have any questions, just attack me, and we try to address the proper way to reach the restaurant and everything.